ahead and start the recording and go ahead and get rolling. Today is Thursday, August 26th. It is our second day of class. We did a lot on the heart anatomy in the last class. So we have a little bit more anatomy to talk about, but a lot of it is going to involve the physiology of the heart and how the heart functions and what the goal of the heart is. And then in lab, we'll talk a little bit more about the cardiovascular anatomy as we talked about at the end of last class. As we were just talking about, you have a handful of assignments that are going to be due over the next 2 weeks. Your lab manuals, again, have 2 components in their reviews, a check your recall and your check your understanding, and you are required to do both of them, and those are graded for time and effort. So make sure you're doing that. You have the 2 Labster activities that are due, so make sure you do that 100% and then get at least 80% correct. And as we talked about in the last class, if you do not get it 80% correct the first time you go through it, you can go through it as many times as necessary until you get the grade that you want. As I also warned you to hopefully stave off some of those emails, it is integrated into Canvas. So like I said, if you complete it at 90% correct, it is only going to give you 18 points for that. But remember, I will then go back in manually and upgrade that to full credit of 20 points. So don't panic if you're only getting 17 or 18 points on there, because I will fix that. But as I also talked about, you have 2 assignments that will be graded for correctness. One is on the interactive physiology, because the ECG is a very important concept that we're going to be talking about today. So I want to make sure we understand those concepts. And it encourages you to take advantage of that interactive physiology. I don't think a lot of people necessarily use that in 430. So it is something that'll be very valuable in this class. And there won't be a tremendous number of assignments. But if you're struggling with concepts, that interactive physiology is a great directed tutorial that can do an excellent job of helping you to understand these concepts. And so by forcing you to do one activity in it, hopefully you will see the benefit of doing others. We have 2 cardiovascular physio-X exercises, 5 and 6. And remember, we're doing 6 first, which has 5 activities, which means you're going to be producing 5 lab reports and turning in those 5 lab reports. Actually, we're doing 3 physio-X's. So the second one is physio-X exercise 5, which has 7 activities. And again, these activities aren't hard, but they're a little time consuming. So I've noticed with some of the assignments that you guys have due, you're waiting to the last minute to get these done. I would warn you that that is not a good habit to get into. Again, technical issues, internet problems, all of those things are not valid excuses for missing assignments or turning assignments in late. So I would encourage you, especially these things that are more time consuming, to get them done sooner rather than later. So there's nothing wrong with turning them in early, right? But if you turn them in 5 minutes late, they're half credit. So it is important to make sure you're turning things in on time, and getting started on them early is a good way to do that. Helps you stay on top of your studying as well, which is important. There is a 3rd physio-X on the blood typing, I believe is what this one is. It is exercise 11, but we're only doing activity 4. So just 1 lab report for that one. And then 1 more unit review and 1 more labster. So that should keep you pretty busy. Also, speaking of busy, hopefully everybody here has already taken the attendance quiz. The access code for that is LOCK. Again, like I warned in the last class, make sure you take that. I did have someone who, like I said, got dropped from my first semester class because they didn't take that attendance quiz. So just pop over to Canvas, take that quiz, let me know that you are here, and we can go from there. All right, questions on any of that?
I have a question. Yesterday I took attendance quiz, so should I take it today too, or is it enough?
Yes, no, this is a different attendance quiz. There was an attendance quiz to show me that you were here on Tuesday. This is an attendance quiz to show me that you're here on Thursday. So you need to take both of them. So just because you took Tuesdays does not mean you do not have to take today's. Everybody needs to take today's to show me that you are here.
Okay, sure.
Yep. All righty. Any other questions? All right, before we dive into the material, we don't have as much introductory stuff, but there are a couple more quick issues that I wanted to go over with you, some kind of administrative things and other things like that. I have not had the opportunity to go through all of the Proctorio quizzes. I know for some of you, you have taken Proctorio before and it's not an issue. And for some of you, this is the very first time. And so in the few that I've been able to grade, and a lot, in fairness, a lot of these notes are based on what I saw from the 430 so far. I haven't gotten to your guys's as much because I'm hoping second semester you guys are a little bit more together than the 430 people are. But there are some important notes that I want to make sure to emphasize with you. First, make sure you scan your entire workspace. That means the wall behind you, so that I know you don't have some plaque or some picture of a human heart that you're using and study off of. Make sure you show your work desk so that I know there aren't any notes or anything along those lines. There are plenty of people who do an amazing job of scanning the room, and I don't quite care what you have on your decor on the across the room from where you're at. I want to know about your workspace. Also, you have to be very careful about your lighting. Obviously, in the computer, you are capable of taking this in the dark, but you're not allowed to take this in the dark. There is a small image on the screen that shows what your camera shows. Now, I know some people don't like that and turn that off, and you're welcome to do that. But if you look in that screen and you see that you are in the dark, or only your forehead is on screen, or also if you have a bright light behind you, you can be backlit, and that can make it hard to see you as well. So you must take the exam with good lighting and keep your head centered on the camera. Again, I appreciate the awkwardness of having to take this at home, and I appreciate that many of you have families and roommates and pets and all sorts of other things that can be distracting. And so I know some people have expressed a desire to be able to use like noise canceling earphones or something like that during the exam to limit the distractions. But unfortunately, there is no way for me to know what you're listening to. And so unfortunately, because of that, you are not allowed to wear earphones during the exam. And then the last thing, again, this is one of those silly things, but I know of 2 students and actually 1 instructor who did this. Remember, you are responsible for showing a picture ID to the camera. Again, it can be your student ID. It doesn't have to be your passport. It doesn't have to be your driver's license, but it just has to have your name and your image on it. And there's that little red square in the center of the screen. The point is to line your ID up with that image on the screen. Don't do what 2 students and 1 instructor that I know did, where they took it and they held their ID up to that square on the screen. Holding it up to the square on the screen doesn't do anything, right? You have to show it to the screen. So again, I know that's a silly thing, but like I said, I know of at least 3 people who did it. So it takes me 30 seconds to mention it, so it is worthwhile. All right, the last thing I wanted to do. So any questions on the Proctorio quiz?
No.
Okay, excellent. Last thing, again, I do read through those student info sheets, and there were a couple things on there that I did want to address. A lot of pre-nursing students, but again, there's still a fair amount of people, a fair amount of diversity of topics that people are interested in, and that always makes the class more fun that way. Also, there's a very diversity of 430 instructors. I would say less than half of the students in this class took me for 430, which is okay. The goal in 430 is to not just be successful in 430, but to gain that information and be prepared. However, I know my teaching style can be different from other instructors. I know some of your classes 430 were completely asynchronous. So there is going to be a bit of a learning curve. The good news with that, as I mentioned, is that there is time to learn. As I talked about, I think I talked about in the last class. If not, then I'll talk about it here. No matter what your grade is on the first exams, you are still capable of getting an A. That means you could get a 0 on the first lab and the first lecture exam, and you could still get an A in this class, mathematically. Now, hopefully nobody's going to do so poorly that they get a 0, right? Because after all, you've completed 430, so you should have some of this information. But I know my expectations can be different. So there is still time to learn, but I encourage you to start right off the bat. As I mentioned, you saw those people who had me for 430. Start a discussion board if you want, where you ask about other students, their opinions of my teaching style and my testing style. and ask for advice and things along those lines. I'm happy and I'm encouraging you to do that because I want you to be successful, right? The material is the material is the material. The femur was the femur, no matter what class you guys took for 430. But how you were challenged on that material is going to be different. And so it's the testing styles that is going to probably be the most different on these. So again, prepare for that, ask questions for that to help you to be successful. Again, most of you were not very non-committal about what you were interested. A lot of people just want to know everything. What I do want to talk about is a handful of you are also taking micro at the same time of this. And what I will remind you about for that is remember that 430 was an incredibly labor-intensive class. 431 is an incredibly labor-intensive class. And microbiology is an incredibly labor-intensive class. And there's only so many hours in the day. Is it possible to take 431 and micro at the same time and be successful and get an A in both of them? Yes, but typically those students who do that don't have anything else going on. If you're working 30 hours a week and you have a significant other and 14 kids and all sorts of other responsibilities outside of school, and you're trying to take micro and this at the same time, then there just clearly aren't enough hours in the day. And again, there's no reason that I should care because I get paid the same whether you get A's or F's, but I know all of you are here because you want to be successful and I want to help you to do that. And so I just want to remind you that for many of the programs that we have listed there, nursing, PA, the sonography programs, those programs are very impacted and grades matter. I know many of you are reaching the end of the classes you have to take and you're feeling the pressure of time or you're anxious to get done and to work your way through and get out of it. But for many of you, there's a big difference in getting an A in this class and getting a B in this class. So I just want to make sure that you think critically about your schedule and whether there's enough hours in the day for you to be able to get the grades that you want in this class. Kind of along the similar lines, for those of you who are retaking 431, especially if you're retaking it with me, but even if you retook it with somebody else, one of the traps that people retaking a class fall into is as I start speaking, they recognize the material and have some familiarity with that material. And familiarity tends to breed complacency. Oh, I remember that. I don't have to pay that much attention, or I don't have to go back and look at that in the book because I remember when they talked about that last time I took the class. What I want to remind you to do is to stay hungry, to stay active, to stay motivated in this class. Right? Remember, you're here retaking 431 because you didn't get the grade you wanted the first time, or you weren't able to complete it the first time for whatever reason. So stay motivated. Don't get complacent. Don't get comfortable with the information. Right? Stay active in the learning process to help you to be successful. All right, study guides. Great question. A couple people asked about this, and let's go back to our modules. As we talked about for the lab exam, the majority of your lab exam are going to come from these types of lab handouts. As I mentioned, this handout that we talked about in the last class that has the blood vessels, has the heart's anatomy, has the microscopy on it. This is your study guide. And remember, as I mentioned, especially for the histology, again, you can look at a histology slide and there are a half dozen easy different questions I could ask. So remember, here in parentheses are the specific terms I will use when talking about these things as a prompt. Right, so that you know exactly the piece of information I am asking for, and you can provide me with that specific piece of information. Remember, as I talked about, people often lose points not because they don't read the questions or know the information, but because they don't read the questions carefully. We did the same thing when we were talking up about here with the blood vessels, right? Yes, you need to know the axillary vein. But remember, as we talked about, there were how many questions I could ask about that axillary vein?
Five.
Five, absolutely. Identify the blood vessel, Identify the blood vessel or vessels that feed blood into it. Identify the blood vessel or vessels that receive blood from it. What direction does it carry the blood, and what is the condition of the blood inside of it? So again, it's very easy when you're, knee deep in the lab exam and you're tired and you're exhausted because you've been up late studying, and to see that arrow pointing at the armpit region of the body and get all excited and go, I know that's the axillary vein. But make sure you read the question carefully to make sure you answer which of those 5 questions that I've asked. So these are your study guides for the lab exam, or at least the anatomy portion of it, right? Because again, you are responsible for all information on all of the exams. Yes, lab exams relate themselves more to identification. But just because I ask you to identify something, can't I also ask you what its function is? Yeah, absolutely. And while essay questions tend to lend themselves more towards essay questions, descriptions, processes, I could ask you to describe things anatomically as well. So you're responsible for all the information on all of the exams. As for the lecture exams, I do not do a review sheet or study guide per se on the lecture material. The reason for that is, as I found, if I give students a study guide, that study guide is all they study. Even though we have all these other resources, all they pay attention to is the study guide. So then if I ask a question that's not on the study guide, students get all angry. And what that means is I basically have to put everything on the study guide. And if I'm writing everything on the study guide, then I'm basically just rewriting my lecture. And I'm not going to be bothered to do that. Right here in the modules, you have the outlines for the lectures that I'm going to be given in class. Now, again, it's not my script. It's not every single word I say, but it is a good indicator of the things we are going to talk about. And the things that we talk about are the things that are going to be important. I'll answer that question in just a second. Let me finish my rant first. As I've said before, one of the things I like about this class is that this class is hard. I don't have to be tricky. As we talked about at the end of the last class, the more we talk about a concept, the more important that concept is, the more likely it's going to show up on the exam. Right? My job in this class is to be a tour guide, right? We don't have to meet back again until Tuesday. So you got a nice long 5-day weekend coming up. Perfect opportunity to go to Paris. And when you go to Paris, do you just start up in the upper right-hand corner of Paris and just go street by street through the entire city of Paris till you reach the other end? Is that how you tour Paris?
No.
No. Instead, you get a tour guide who takes you to the 12 most important places. And that's my job. My job is to take the textbook and distill it down into the most important concepts. So if there's a concept in your textbook that we don't talk about, you don't have a homework assignment on or anything like that, then it is not going to be on the exam. The only rarely are there instances will be otherwise. Probably the best example is actually back in 430. In 430, the tissues and the integumentary system tends to always run long. So at the very end of the integumentary system, sometimes I run out of time and I have to say to the class, all right, you guys need to go to the textbook and you need to read the part where it talks about burns, because we're not having time to talk about it in class anymore. So read about burns because burns are going to be on the exam. Right. And I specifically tell you that. But if we don't talk about burns in class, there's no homework assignments on burns, then burns aren't going to be on the exam. So these outlines are not an alternative to coming to class, but they should help you to remember and emphasize the important concepts we are covering in class. Now, labeling activities, to answer Claudia's question, are primarily just on the lab exam. But like I said, could I point at something and also ask you how it functions? Again, most of my examples relate better to 430. I could show you a picture of a cell and point at the Golgi apparatus and ask you to identify it. That's a very obvious lab exam type question. But on the lab exam, could I also point at the Golgi apparatus and ask you its function, what it does in the cell? Yeah, absolutely. This is one class. We don't have a lab class and a lecture class. Lab and lecture are really different modalities for studying information. And because we're 100% online, that line's blurred even more, right? Most of you got through 430 and you never held a bone in your hand, right? So even more than before, it's all the same, right? It's just different ways of covering the material. So you are responsible for all of the material on all of the exams. And that's why we have the exams on the same day, because you're responsible for the same chunk of information, and then show me what you know on the lab exam and show me what you know on the lecture exam. All right.
Hey, Professor.
Yes.
Are you going to be doing the lab the way that you did the first one from 4:30, where it's all on one? Are we going to try that again?
I'm not sure what you mean by all on one.
So it's all on the same page as opposed to loading every individual?
Oh, no. Great question. Unfortunately, again, I have for the past 2 semesters, both spring and then also in summer, I attempted the first test to, because I prefer all the questions be present at once for the online, but for whatever reason that doesn't appear to work. And so what tends to be most compatible with most people's systems is to present the questions one at a time. And so, and honestly, that's how we do it in the classroom. So I'm not hating the fact that we do it that way, but I don't prefer it, but that is the way that seems to work best. So yes, for the lab exam, 1 question at a time. For the lecture exam, you'll have all the questions at once. Yeah. Now, again, with the 1 at a time, you can go back to previous questions. So if you're question 15, and when you look at that image, you realize you had 2 wrong, you can go back to 2 and change it. So it's not like once you write an answer, you have to stick with that forever. You can go back and change it. But if you're bouncing around a lot, that does cut into your time for the exam.
Professor?
Yes.
Quick question on the so on the syllabus, there's the schedule, right? And then whatever day it may be, we'll have exam 1. And on that day, we have both lecture and lab exams, correct?
Correct.
And then on the exams, do you give us like 5 minutes or something to kind of go through the scanning process, or if we're having a little trouble, or is it just like, yeah, you have to start at 12 and so on?
So great question. The way this works, I don't even think the exams are up. I know the exams aren't up. Let me rephrase that. Our class time is from 12 to 4:35. All right. During that time, you must complete both exams. All right. Don't have to start it during those times. You have to complete them during those times. So you're right. Right at 12 o'clock, you can start the exam. Now, there are 2 exams. There is a lab exam and there's a lecture exam. The lab exam, it varies tremendously. My guess is somewhere between 60 and 90 questions, somewhere within that range. The cardiovascular system has a lot of anatomy, so that one's probably going to be closer to the 60 range. Our second unit, where we're doing endocrine and lymphatic, has a lot less anatomy, so that'll probably be closer to the 60 range, but it'll vary from test to test. Typically, you will have about 1 minute per question. Plus, you're right. Where's my question mark? Can I find my question mark? Oh, there it is. There. Per question, plus about 20 minutes procedural time. So that means, the time to be able to scan your area, the time to skip from one question to the next, because again, these are going to be 1 question at a time. So that's usually about an hour to 90 minutes. Somewhere in that, not 160, from about 60 to 90 minutes, depending on somewhere where it is, somewhere around that range for that. The lecture exams are usually somewhere between 25 and 35 multiple choice questions. Usually somewhere between 2 and 6 fill in the blank questions, and usually somewhere between 6 and 9, let's say, essay questions. And for that one, depending on the size, you usually have about 2 to 2.5 hours. Again, that's the time you have to take it. Most people don't use the full time to take the lecture exams, but that's the time that's available. So notice with this, you have somewhere around 3 to 4 hours worth of exams to take on that day in a 4.5 hours period of time. You can take the exams in any order that you want. However, I strongly recommend you start early at noon or as close to it as you can and start with the lab exam. I would say 90% of the problems that people have with their exams occur with the lab exams. So if you start with the lab exam first and you don't have any problems and you breeze through it, winner. You can take a little bit of an extra break before you start your lecture exam. But if you start early and you do have problems, then we have time to fix it, where you can then resolve that, take the exam, and then still have plenty of time for the lecture exam afterward. So you're not required to take the lab exam first. If you're really jonesing to take the lecture exam and you want to do that one first because you're worried about how much time it'll take because it's a longer test, fine, you can do that.
Got it. Thank you, Professor. And then I have one more question for your essays, because I know some professors are different. What is the highest point question that you will ask for one of your essays? Because I know some professors, they'll make like a 15-point essay question and have you like write like 2 paragraphs.
It's a great question. I don't know. My guess is probably, I mean, there's no set point value for the essay question. The more information on the question, the more points it's going to be worth.
Right.
I think I've had questions worth 10 or 12 points, but I've also had essay questions worth 4 points. So, I mean, it really depends more on the question than anything else.
Got it.
And as far as the other part of what I wanted to address, what you said paragraphs, and that is fine. If it takes you 2 paragraphs to answer the question, that's awesome. But again, be a smart, sophisticated student. If it's a 4-point question, don't write 2 paragraphs, right? That's clearly way too much information. But I don't even care if they're paragraphs. All I care about is the information. I want the information. And I want the details. I want the specifics. If you organize better by bullet points, that's fine. However, what I warn you is often when people use bullet points, they tend to be more brief in their information, and that's going to lose you points. I don't care about your format. I care about the information. When we're giving this exam in person and I'm using paper to give them, I usually go through about 1.5 red pens, writing describe, explain, be specific, and I want details, right? That's the other half of that tour guide. If I'm going to take the 20 concepts in chapter 16 and I'm going to bring them down to just the 12 most important, then you have to own those important concepts. I want specifics. I want details. We talked about it in the last class when we were looking at that histology slide, and I asked to identify the tissue. Muscle is correct, but remember, cardiac muscle was a more specific and a better answer. And that's what I'm looking for. I'm looking for specificity. I'm looking for details. That's how you show me you've mastered the information, is by being specific. Don't just tell me that sodium enters the cell. Tell me sodium enters the cell through a chemically gated cation channel, right? I want details.
Got it. Thank you.
Yep. Excellent questions. Any others?
Question. So how do you take into account breaks during exam period? And second question is, if we have questions regarding a specific question, do you have a separate room where we can go and have a discussion?
Nope. Unfortunately, there is no way to interact during the exam. So again, I put a tremendous amount of time and effort into the questions to try to make them as precise and clear in what it is that I'm looking for as possible. If you are confused, you can write that in your answer. I'm confused by this aspect of the question. If you mean this, then this would be my answer. If you mean that, this would be my answer. You could do something like that, but that'd be probably about the closest you could get to something like that. As far as breaks during the exams, there are no breaks during the exams. With 2 exams, though, as you can see, that they're not going to take the whole class time. So if you take your first test at the beginning of class, then you have time to take a break before you do the second one. And in fact, I encourage you to not just go straight from one exam to the other. I encourage you to take a break in between, take a biology break, get something to eat, get something to drink, more caffeine, stretch, right? Relax your brain, all of those things. But once you start the exam, the test, the timer's going and it must be completed. And again, as I mentioned, it must be completed during the class time. If you start the lecture exam at 4 o'clock, you're only getting 35 minutes to complete that lecture exam. And I can guarantee you that's not enough time to finish your lecture exam. So like I said, you're not starting it during class time. You need to make sure that you finish it during the class time. So plan accordingly. And there you go, Amanda. She mentioned usually at about 20, 30 minute break between the exams last semester. So there you go. So if you take one, especially if you take it at the beginning of the class, and especially this class, my other class meets at 8 o'clock in the morning. If you really, really hate getting up early and you want to get up at 8:30 instead to start the exam, bonus. We start at noon. There's no reason not to be ready to go at noon, start the exam, and then see how long it takes you. And then from there, plan accordingly for your break. Yeah, 20 to 30 minutes seems like an excellent break in between there. All right. Excellent. And like I said, I purposely give a lot of time for the lecture exam because I feel people feel the pressure of time on those things. Most people, I would say, finish the lecture exam under 2 hours, usually about an hour and a half. So you typically have far more time than you need, but especially there are some people who are very long test takers, really like to take time to do their critical thinking and their brainstorming and stuff like that. And I want to give those people the opportunity on the lecture exams to be successful. Lab exams, whether they're in the classroom, whether they're online, they're always timed, they're always tight. That's always, you need to be ready for that recall. And you'll find, when we're doing a timed exam in the classroom, a full minute for a question is a long period of time. So it's one picture, one question, and you should have plenty of time to be able to do that as well, if you're prepared. All right. Awesome questions. Any others? All right, spectacular. That brings us back to the last bit of our study guide. There are a couple questions for me. I have been teaching at American River College for this is, I think, like my 15th year now. I've been teaching anatomy and physiology for over 20 years. I started teaching at Davis. I got my both undergraduate and graduate degree at UC Davis. So I cut my teeth in Davis for a very, very long time. Where I got my PhD in neuroscience, studying the development of the visual system. But I tremendously enjoy teaching. Yeah, for those of you who are familiar with research, doing research is amazing. It's like walking on the moon. It's an amazing experience to do those types of experiments. I worked in a comparative neuroanatomy lab, and it was a tremendous amount of fun. You never knew what was going to be on the dissection tray every time you came in. But research is very much driven by grants, and I was not interested in sitting around and writing grants all day long. And I tremendously enjoy teaching, but I should say I enjoy teaching this period. I love being at college. I love teaching anatomy and physiology. This is not a required class, right? This isn't a GE that everybody has to take no matter what. The people that are here, like you, are motivated. They have a goal. They have expectations, and it is an amazing class to teach, and I enjoy it tremendously. Someone asked me what the hardest section is. I think the hardest section in all of anatomy and physiology is probably the second section of 430, when we do the histology and we do the tissues and we do the skin, because it's the part that's most unfamiliar. In 431, I would say it's probably either this section, because someone who didn't have me for 430 maybe had someone who wasn't quite as vigorous of a 430 instructor, might underestimate the workload that is going to be responsible for them. So the averages tend to sometimes have a wider range on this first exam. But the second section, I think that's the one where we're doing the lymphatic and endocrine system together, where we're talking about hormones and all of their interactions. And we're talking about our immune response and all of those interactions, chemical and cellular things, right? The fun thing about this section is you can hold a heart in your hand. You can physically look at blood vessels. You can't look at a T cell in your hand. It doesn't work the same way. So I think probably our second section is the one that is least intuitive. And because it's least intuitive, I think that's the one that makes it a little bit harder. Although I think having it online does actually help in that format. Right? When you don't have the heart models to hold in your hand, it makes it a little bit more challenging because you don't get that 3D representation, but there aren't 3D representations really in the lymphatic and endocrine system. So in some ways, I think it may make it a little bit easier. All right, so that was the last and the least of the information. Questions on any of this stuff or anything else that we've talked about so far?
Yeah, Professor, I had a question.
Yes.
So if we do decide to come on campus for the open lab, how will the vaccines be checked? Should we bring a vaccine card or?
That is an excellent question. My understanding is the vaccine requirement is a requirement for all students, regardless of their format. So even if all the classes you're taking are online, I think technically to still be a student at Los Rios, you must be vaccinated. So all of the vaccination issues, I believed, are handled through admissions and e-services. So the expectation then when you come on campus, if you're enrolled in a class, is that you are vaccinated. So it is my understanding that it is not Jeff's responsibility to be checking people's vaccination cards. Again, I'm guessing on this, but I think it's a fairly educated guess. What I do know is that, well, yes, there's on e-services, you have to submit it. There's a spot where you submit it. What they haven't made clear is what they're going to do to students who haven't completed that. At least to us, they haven't made that clear. I don't know if admissions has already made a decision as to whether those people will automatically be dropped or what's going to happen. They have not told us as instructors, but I'm going to go under the assumption that if you haven't had the vaccination, you won't be able to participate. And so therefore it would be a moot point as to coming on campus. Jeff's responsible for making sure that you wear your mask in the classroom. So that's the primary responsibility that he has. Okay.
Thank you.
Which is quite frankly something we're used to doing anyway. All right. Any other questions? All right, spectacular. With that, we are done with all of our introductory stuff. That took a little longer, but that's okay because we covered some good, important stuff. So I like that. I appreciate that. All right, I don't know who's driving, but I'm going to go ahead and mute everybody. Please feel free to unmute to answer questions when we get to that. All right, we left off last way. And like I said, we had talked about a lot of the gross anatomy of the heart and how basically there are these two independent but related pumps that are responsible for delivering blood to the body. And of course, with two pumps, we have how many circulations of the body?
3.
3, right? How does that work? How do we have 2 pumps but 3 circulations? Well, we do. So let's see if we can figure out what they are. Give me one of the circulations.
Pulmonary.
Excellent. What is the permeant? What is the path of the pulmonary circulation?
Starting from where?
Wherever you choose. I guess the heart, since the heart's the one doing the pumping.
To the lungs, and
There you go.
then back to the heart.
Excellent. From the heart to the lungs and back to the heart. Excellent. Why? What's its function?
To reoxygenate. or to make the blood more oxygen rich?
Do we just care about oxygen?
No, the carbon dioxide.
Yeah, so the function is indeed gas exchange. And you are correct to increase oxygen levels in the blood, but we can't forget about how it's also involved in decreasing CO2 levels in the blood. And what structures are involved? Well, if it leaves the heart, where does it leave the heart from?
The
Right
pulmonary
ventricle.
arteries?
It's going to go into the pulmonary trunk and then the pulmonary arteries, but where does it leave the heart?
The right ventricle.
Right
There we
ventricle.
go. From the right ventricle into the pulmonary trunk and arteries, right, to the lungs, gas exchange in a capillary, pulmonary veins back to what, a final structure of the heart?
Left atrium?
Left ventricle, there we go. Left ventricle?
Left atrium.
Atrium.
Atrium, left atrium. Excellent. Perfect. There you go. Excellent. What's the second circulation?
Systemic.
Systemic, oops, circulation. So let's do the same thing. From the heart to,
The body tissues.
yeah, the body. I like that. Self-built tissues, right? Excellent. And then back to the heart. What is its function?
To supply oxygen and nutrient. to the body.
Absolutely. We absolutely are going to do more gas exchange again, but also nutrient and waste exchange. Right? Basically, exchange of materials.
Does it also keep the body warm, temperature regulation?
Sure, but this is also, yeah, water, temperature, all those other things we talked about being transported, chemical signals. All those things that need to get to the tissue. So let's actually generalize it that way. The function is to get stuff to the tissues that the tissues need and take stuff away from the tissues that they don't need. That's a pretty general. Oh, where did that go writing? Take stuff away from. It's a pretty general definition, but not a bad one. Serves our purposes. Excellent. What are the structures involved?
Left ventricle.
Excellent. And let's just do it this way. Systemic arteries, two caps, to systemic veins, and most importantly, where does it come back to the heart?
Right atrium.
To the right atrium. Perfect. Questions on that one? So then what's left? How is there 3 circulations? What is the third circulation?
Coronary.
There we go. What's the job of the coronary circulation?
Blood vessels that supply the heart.
Yeah, exactly. I like that.
The
Basically
myocardium.
from the heart to the heart, or really to the heart muscle. And then back to the heart again. Absolutely. As we talked about, your heart is only about the size of your fist. However, it has a massive blood flow to it. It's like 1/20th or 1/200th of your body size or something like that. I've got the numbers in here when we'll look at this in a second, but it has a massive, massive supply of blood to it because, as we talked about, it is constantly active. It is highly reliant on oxygen, and if it's deprived of oxygen for even a brief period of time, that tissue can be damaged. But that's okay because we can repair tissue, right?
Not cardiac.
Not the heart muscle, absolutely. So being non-regenerative, it is vitally important to make sure that that heart muscle gets the oxygen that it needs to function. And that is indeed its function. Supply oxygen, yes, and obviously the nutrients and things like that to the heart walls. Excellent. So then what that leaves us with is the structures. The blood going to the wall of the heart, where would it begin?
Aorta.
Yeah, well, okay, but it's got to start in the heart. So if it's going to go into the aorta, where does it start?
Left ventricle.
Excellent. From the left ventricle, it goes into, and again, aorta is 100% correct, but remember, we want to be more specific. So let's be more specific. Into what?
Ascending aorta.
Remember, one of the things we talked about is one of the reasons we are dividing the aorta into 4 distinct regions is those 4 distinct regions have specific blood vessels that are responsible for sending blood to specific parts of the body. And the ascending aorta only has 2 blood vessels coming off of them. Anybody know what the 2 blood vessels coming off of the ascending aorta are? Sorry, Amanda, you're breaking up. I can see you're trying to talk, but I'm not able to hear you very well. Close. Remember, the vena cava is the one coming back. Those are the ones that feed into it. What are the 2 arteries that come off of the ascending aorta?
The left and right coronary arteries.
Exactly, the left and right coronary arteries. Notice the very first thing that gets that oxygen-rich blood as it leaves the heart is the heart itself. So the only 2 big blood vessels coming off the ascending aorta are the right and left coronary arteries. These feed into the blood vessels and the capillaries of the heart. And then it needs to return. Let's start easy. What part of the heart does it need to return into?
The left and right coronary veins.
Well, we'll talk about the veins in a second. No, but which of the 4 chambers of the heart, after the heart muscle is done with it, do we want it to go into?
Right atrium.
So what were the blood vessels we've identified going into the right atrium?
Superior, inferior vena cava.
So which one does the heart use?
Superior.
No, that's the third one you said.
Exactly. Neither is the right answer, and Aravec is correct. Remember, I told you that there are actually three major blood vessels that feed into the right atrium. Superior vena cava is one of them, inferior vena cava is the other, and the third, as Ala has pointed out, is indeed the coronary sinus. So the coronary sinus is what collects the blood from the heart and deposits it into the right atrium. We've done this all with the pretty words, but let's take a look at the pretty pictures that help us to understand this. Here we see one of the pretty pictures that does a good job of showing us two of our circulations. Notice here is the systemic out from the left ventricle into the ascending aorta and throughout all of the body. The systemic circulation contains the majority of the blood vessels in the body. And so notice, as we've talked about, this is why we say that most arteries are oxygen rich and most veins are oxygen poor, because most arteries and most veins are systemic, and that's their function. However, remember, most isn't all, and that's because our pulmonary circulation is the one that screws it up for everybody else. Notice in our pulmonary circulation, the arteries moving away are oxygen poor, and the veins are oxygen rich. And so we see that oxygen poor blood being pumped out of the right ventricle into the pulmonary trunk, pulmonary arteries to the lungs for that gas exchange, and then back to the heart. And here's another pretty picture that shows all of that as well. But as we've also talked about, there is a third circulation, the coronary circulation. As I mentioned, the heart only makes it about 1/20th of our body weight, but it receives 10 times as much blood, 1/20th of our blood supply. And notice, as we look at this illustration here, here is that ascending aorta. That's a horrible color. That ascending aorta. And notice there's only two blood vessels that come off of it, the right coronary artery and the left coronary artery, to feed the walls of the heart. Yes, the heart is filled with blood, but the muscular wall is so thick, it can't get all of the oxygen that it needs just from diffusion. So it has a big, elaborate, interconnected blood supply that all feeds into this big coronary sinus at the back of the heart, which then feeds it right into the right atrium. Yes, I can go back, Jane. Hold on a second. So we need to get rid of that and that and that and that and that. Oops, too far. And now I think I can bring it back. Oh. Was I doing that on the whiteboard? I thought I did that here. Do I need to go back further? Sorry, apparently I don't seem to be able to go back to those notes. I apologize, Jane.
It's okay. Thank you.
All righty. Questions on that? All right, so 2 pumps, 3 circulations. Excellent. Now, in our last class, we also spent some time talking about the microscopic anatomy of our cardiac muscle. We talked about how it is similar to skeletal muscle in that it is striated. But like we learned in 430, we can now be more sophisticated. What does it actually mean to be striated? What is the indication? What makes it striated?
Sarcomeres.
Yes, it contains sarcomeres. Sarcomeres, remember, are those functional and structural units that allowed skeletal muscle to contract. And of course, we know that they are arranged in a row to form a special organelle known as the organelles formed by sarcomeres.
Vesicles.
No,
Myofibril.
there we go.
What's that?
Myofibrils, absolutely. Notice our illustration down here does a decent job of showing this, showing all of these myofibrils lining up like ducks in a row inside of our cell. And here we see many sarcomeres in a row that way. The nice thing is we learned everything you wanted to know about sarcomeres and myofibrils in 430. So the good news is the process of contraction is very similar in cardiac muscle to a skeletal muscle. Of course, there's that pesky word similar, not identical. And while it has those myofibrils, it has those sarcomeres, it has those stripes, it's not identical to skeletal in that it is short, branching shaped cells. They have those special intercalated discs that connect it from one cell to another. And notice, conveniently enough, on our illustration, it shows how those intercalated discs contain gap junctions for the rapid diffusion of ions and desmosomes to lock everything together. Unlike skeletal muscle, which is completely voluntary, cardiac muscle is involuntary, meaning that what controls it?
The autonomic nervous system.
Anything else?
Hormones.
There we go. So hormones in the autonomic nervous system are going to be able to modify the behavior of it. But notice also, as we look at these illustrations, there are some other dissimilarities. Notice, like in skeletal muscle, there are transverse, blue is a horrible color for that. There are transverse tubules that penetrate deep down and wrap around the sarcomeres, bringing the interstitial fluid deep into the muscle cell. Notice our myofibrils are surrounded by the sarcoplasmic reticulum. What did the sarcoplasmic reticulum do again?
Release calcium.
Store and release calcium, absolutely. And remember, in skeletal muscle, there's a huge enlargement of the sarcoplasmic reticulum right next to that transverse tubule. What did we call that?
Terminal cisternae.
Terminal cistern, and the terminal cisternae on one side and the terminal cisternae on the other side, and the transverse tubule in the middle together formed a tetrad. But notice there are no tetrads here. Yes, there's sarcoplasmic reticulum, but it's relatively scant, it's relatively small, and there's no big enlargements. One of the main reasons for this is cardiac muscle relies much more on outside calcium. Remember, basically, skeletal muscle has all the calcium it needs to move the regulatory proteins. That is not true for cardiac muscle. Cardiac muscle relies much more on calcium from outside of the cell to move those regulatory proteins. And like I said, they're incredibly dependent How are we on time? They're critically dependent on oxygen. So there are many, many mitochondria because we want to be able to use that oxygen, make that ATP as efficiently as possible. Because as we know, lactic acid from producing ATP anaerobically can be very toxic and damaging to a muscle cell. So we want to limit that as much as possible. We want to get massive amounts of oxygen in here, have tons of mitochondria to, as efficiently as possible, make as much ATP as we can. All right, questions on that? All right, excellent. Then what we are going to do now is we're going to scratch some of those itches. We learned, like I said, a tremendous amount about skeletal muscle tissue and how it works. And so we're going to relate that information we know and recall about skeletal muscle tissue to how it is similar, but also different from cardiac muscle. Now, we didn't get quite as far in the lecture as I would have liked, but I think this is important information and it leads us into even more and more interesting things. So I think this is a good natural stopping point. Let's go ahead and take our first break here. Again, if you have not taken your the attendance quiz, please let me know. Please take it now. Was it lock? Was that the code? Yeah, so the access code is lock. If you have not taken the attendance, please take that so that I do not drop you from the class. All right, let's go ahead and take our first break. It is 1:03, so that means we will come back at 1:18, and at 1:18 we will restart, and I will start the recording at that point. All right, any questions before we take our first break? All right, see you in 15 minutes. Let's go ahead and get started. Any questions? All right, so as I mentioned, there are definitely some similarities in how a skeletal muscle and a cardiac muscle contract, but there are also some big differences as well. For starters, there are no motor units in cardiac muscle. Someone remind me what a motor unit is?
Is it the motor neuron plus all the muscle fibers it controls?
Yeah, absolutely. It is the somatic motor neuron. And all of the skeletal muscle fibers or cells it attaches to, or it synapses on, let's say it that way. And remember, they all fire together. So when that one somatic motor neuron fires, all of the skeletal muscle cells that it's connecting to all stimulate, all contract, all relax at the same time. And remember, these motor units can be as few as 2 or 3 muscle cells to as many as thousands together. Well, don't we want the heart muscle to be working together in a uniform fashion? Why don't we need motor units to coordinate the activity of the cardiac muscle?
Well, aren't they firing like at different times? Like one part of the muscle relaxes while the other one fires?
Too, but we do still want some coordination in these muscle cells. You're right, we want to direct that signal, but we still want some coordination. And why aren't we using motor units for that coordination?
It's involuntary?
No, that is true, it is involuntary, it is true it has a longer contraction, but that's not why we don't need motor units. Remember, motor unit gets all muscle cells to work together as one. Why don't we need motor units to get cardiac muscle cells to work together as one?
You risk damaging the heart.
No, we want the muscle cells to work together as one. We just do it in a different way. You guys are overthinking this.
Is it through a wave summation?
Nope. Well, eventually we're going to want to contract every muscle and we're going to want to coordinate them together. If only there was something, say, in between cardiac muscle cells that allowed ions to freely flow from one to another so that they work together as one instead of requiring some type of motor unit to do that.
Gap junctions.
There you go. Absolutely, right? Our cardiac muscle cells have gap junctions. Those gap junctions within the intercalated discs are what allow the cardiac muscle cells to coordinate their activity. So we don't need motor units to coordinate their activity. Gap junctions coordinate their activity instead. Now, you guys hit on some other big differences between cardiac and skeletal muscle. The length of the contraction is dramatically different. Remember, cardiac muscle cells can be as fast as 20 milliseconds to as long as 100 milliseconds for the contraction. But a heart muscle's contraction is over 200 milliseconds. Why do we want a much longer contraction in our cardiac muscle cells?
You pump more blood?
Right, it makes it a more efficient pump. Right? If you've ever pumped up an air mattress before, and I don't mean the way you do it now, where you plug it into the wall and you push a button and it fills up. No, I mean with a hand pump. When you're using that hand pump, you don't want a bunch of really fast, rapid, short pumps of it. To really efficiently move the air, you need a nice, long, sustained pumping of that. And it's the same thing with the heart. To efficiently pump blood, we want a longer, sustained contraction. Someone remind me what the absolute refractory period was again?
A period in time after a muscle has contracted where no amount of stimulus will allow it to contract again?
Absolutely. Basically, it's an amount of time where the cell loses its excitability. Right? It cannot be stimulated again. To contract. Now, how long was that absolute refractory period in a skeletal muscle?
Was it around 4 milliseconds?
Yeah, or less, really. It's like 1 to 2, maybe 1 to 5 milliseconds. It was incredibly fast. I think Jacob mentioned it earlier. One of the advantages of that is we can stimulate it very rapidly, produce a lot of twitches, add all those twitches together to get that tetanized contraction of the muscle cell using that wave summation. However, notice the absolute refractory period for our cardiac muscle is actually longer than the contraction. Are we going to be able to wave summate cardiac muscle? Are we going to be able to add waves of tension together if the absolute refractory period is longer than the length of the contraction?
No.
No. Which means we can't tetanize the muscle cell. And doesn't that actually sound like a good thing? Would we want to tetanize, lock up our cardiac muscle in a sustained contraction where it can't let go?
Sure hope not.
No, we wouldn't want that to happen. Absolutely. So it's actually a good thing that it has such a long refractory period. And remember, there are 3 main types of skeletal muscle tissue, right? Fast glycolytic, right? Slow oxidative, and fast oxidative. Well, it turns out there are 2 types of cardiac muscle. The first of these, the most common type, is our contractile cells. So we have cardiac contractile cells. And guess what cardiac contractile cells do?
Contract.
Contract. See how easy that is? Absolutely. And they make up about 99% of the cardiac muscle cells. But there's one other thing about cardiac muscle that I don't think we've mentioned. I think one person mentioned it one time, but we haven't talked about it enough. Remember, if I take my arm and rip it off and set it on the counter and provide it with all the oxygen and nutrients it needs, is it going to beat on its own?
No.
If I rip out my small intestine and put it on the counter, is the smooth muscle on there going to contract on its own?
No.
No. What if I rip my heart out, put my heart in a beaker, give it all the oxygen and nutrients that it needs? is it going to contract on its own?
No.
Yeah.
Yeah, because remember, cardiac muscle, 1% of the cells, just a very small amount, but a small amount of cardiac muscle cells are highly specialized cells that are known as conduction cells or known as autorhythmic cells. And those autorhythmic cells are able to produce their own action potentials. And it is them that drives the basal starting rate of this heart. Yes, we can use autonomic nervous system and we can use our hormones to modify it, make it beat faster or make her beat slower. You're doing it right now as we speak. Because if I ripped your heart out, put it in a beaker and left it alone, it would actually beat at a rate of about 100 beats per minute. Is that what your heart rate is right now while you're sitting in your class?
No.
No, unless you had a quad shot from Dutch Brothers. Hopefully your heart's not going 100 beats a minute. So our autonomic nervous system, our parasympathetic nervous system, is actually suppressing it while you're sitting here calmly in class. So not surprisingly, two very different cells with two very different functions. And we're going to have to talk about both of them. But let's start with the contractile cells, because I think those are going to be easier for us to get our head around, because basically they're the ones that are most similar to skeletal muscle, which is something we understand. Let's start first with the muscle action potential of our skeletal muscle cell. Now, of course, if we're going to do this, we're going to need a graph. And anytime we have a graph, we need to label our axes. So this, of course, is going to be time. This over here is going to be voltage. And we get to remind ourselves of some numbers here. If we take that skeletal muscle and put it in a petri dish and provide it with all the oxygen and nutrients that it needs, right? Its membrane potential is going to happily stay the same forever. What do we call that point again?
Resting membrane.
Resting membrane potential.
Excellent. And as long as we leave that cell alone, it'll stay at its resting membrane potential forever. And anyone remember just off the top of their head what an average resting membrane potential is?
Negative 70 millivolts.
Yeah, about negative 70 millivolts is a good number for that. Excellent. So as long as you leave that skeletal muscle cell alone, it will happily stay at its resting membrane potential forever. However, as we learned in 430, if we disturb it, if we depolarize it enough, we will reach threshold and we will get a very rapid depolarization. And what ion is primarily responsible for that rapid depolarization?
Sodium.
Yeah. There is a massive influx in sodium because sodium is trying to get to its resting membrane potential. And say we have a huge, massive influx. But just as rapidly as we have that rapid depolarization, the cell just as quickly goes back to its resting membrane potential. It has a rapid repolarization. Notice it doesn't quite have the undershoot the same way that a neuron did. It's pretty much just going to go to its resting membrane potential. But like a neuron, what ion is primarily responsible for the repolarization?
Potassium.
Yeah, leaving. So the efflux of potassium out of the cell causes it to just as rapidly repolarize, and then it's back to rest. And as we talked about, this action potential takes somewhere on the order of 1 or 2 milliseconds. It is an incredibly rapid, almost instantaneous process. And we learned one more thing back in 430. We learned that one neural action potential produces one muscle action potential, and that muscle action potential produced an increase and decrease, or more specifically, an activation, increase and decrease of tension in that muscle cell. And what did that we call that?
Twitch.
There you go, one twitch. Absolutely. So the other piece of information we can put up here is the twitch of a skeletal muscle cell. Again, we can put our graph on the bottom here. We, of course, have time. And now over here, we have our tension. Of course, as we know, tension starts at 0. The cell stays at 0 for a while. Until a stimulus is produced. This point right here, we produce our muscle action potential. That is our 0 point of time. And that twitch has how many stages, how many phases to it?
3.
3. Excellent. What is the first stage called? Excellent. Latent period. Oops. Ugh. Our latent, God, I did it again. Our latent period. And how does tension change during the latent period?
It doesn't.
It doesn't. So the tension stays at 0. Excellent. What's the second phase? The second phase of a twitch. Heck, latent was the hard one. There we go. Contractile period. And how does tension change during the contractile period?
It increases.
We get an increase. Excellent. And then it finishes with the third phase, which is what? Relaxation. And how does the tension change in that relaxation period?
Decrease.
Decreases until the cell is back to 0 tension, at which point our twitch ends. And as we mentioned, these twitches, they last anywhere from 20 to 100 milliseconds. So again, despite the lack of enthusiastic responses, there is no new information here on the board. This is all stuff you should have learned in 430. But I'll ask anyway. Any questions on any of this? All right, excellent. Then let's compare what we know with what is new. And so first we are going to talk about, again, we are specifically talking about contractile cardiac muscle cells. And we will talk about their muscle action potentials. I'm going to go ahead and abbreviate it there because I wrote it out on the other side. And again, we are going to draw a graph. And again, this graph will involve time versus voltage. Again, we are going to have a resting membrane potential. And if we leave this contractile cell alone in its petri dish, what's going to happen to it?
It's gonna contract.
Turns out, no, that is definitely a good guess, but it turns out, no, if you leave this cell alone in a petri dish, much like our skeletal muscle cell, it will happily sit at its resting membrane potential forever. Much like skeletal muscle cell, which when stimulated contracts and produces tension, our contractile cardiac muscle cells only contract when stimulated. So when stimulated, when there is a stimulation, so let's actually add that to remind us. Stimulation. Stimulation. When there is a stimulation, just like we see in our skeletal muscle cell, we see a rapid depolarization. And what do you think causes that rapid depolarization?
Influx of sodium.
Yeah, once again, it is a sodium influx. Massive amount of sodium enters into the cell, and we get that rapid depolarization, just like we saw in our skeletal muscle. But then something different happens. Yes, just like in our skeletal muscle, sodium stops entering the cell, just like it does in our skeletal muscle. But two other things happen as well. Normally, notice we saw a rapid influx of potassium because potassium channels opened. What actually happens here is the permeability of potassium decreases. That's a fancy way of saying less potassium can leave the cell. And if less potassium can leave the cell, if less positive ions leave the cell, then doesn't that leave the inside of the cell more positive?
Yeah.
Yeah.
Yes.
And one more thing happens. voltage gated calcium channels open. And when calcium channels open, which way does calcium move?
In.
So notice no more sodium is coming in, but calcium is now coming in. Remember, I said cardiac muscle was much more reliant on this extracellular calcium to move the regulatory proteins. And less potassium is leaving. So in our cardiac muscle, what this causes is a prolonged depolarization. And that prolonged depolarization is a period of time we refer to as the plateau stage. Now, eventually, those potassium channels are going to open. And we have an efflux of potassium. We don't need to worry about a threshold on this. We just know that there's a stimulation enough to cause and trigger this, this effect. We get this efflux of potassium, and that causes the cell to repolarize. Notice this plateau stage produces a much more prolonged action potential. This action potential can be, oops, whoa. This action potential can be 15 times greater, longer than skeletal muscle. It is a much, much longer action potential. So not surprisingly, a longer depolarization is going to produce a longer twitch. And so that's what we can look at next.
And what causes the potassium to be less permeable?
There are specific potassium channels that close during that period of time, and you don't need to know what those channels are or why they close. Just know that the permeability decreases. So again, it's going to start the same way. And it has the same 3 stages. So of course, we start out producing no tension at all. There is a stimulation where we produce the muscle action potential. And when that muscle action potential is produced, we get a latent phase we get a contractile phase, and we get a relaxation phase. So we still have those three phases. But the difference here is the timing. As we mentioned, this entire twitch lasts for over 200 milliseconds. So it's got the same three phases, just with a longer depolarization, we get that much longer twitch, which, as we talked about, produces that prolonged contraction to efficiently pump the blood. And then we'll layer one more thing onto here. Refractory period. Remember, the absolute refractory period is done well before our skeletal muscle is finished with its twitch. That's how we're able to add those waves of tension together, get that wave summation, and tetanize the muscle. Whereas, remember, our absolute refractory period for our cardiac muscle is after the twitch ends. So no wave summation, no tetanizing of the muscle. Questions on this? This doesn't make sense. It's only going to get worse from here. So if you have any questions, now's the time to ask it. All right. Again, summarize. Here's our skeletal muscle action potential. Here's our skeletal muscle twitch, and those absolute and relative refractory periods. Right. Whereas, again, much, much longer for those refractory periods for our cardiac muscle with that prolonged action potential. Starts with the depolarization of the sodium channels. Calcium enters the cell, maintaining that plateau stage, as well as the decrease in potassium levels. But eventually it repolarizes as those voltage gated potassium channels open. And our calcium leaves. And again, because of that, we produce a contraction that is much, much longer than the contraction produced by skeletal muscle. So I'll ask again, any questions on this? Because like I said, if this doesn't make sense, it's just going to get worse from here. So, not surprisingly, with some variations, contractile cells in the heart do pretty much the same thing that contractile cells in the arm do. They get stimulated, they produce an action potential, and they contract. And when they're not stimulated, they happily sit at the resting membrane potential, waiting to be stimulated. However, the biggest difference between your heart and your arm is not what happens with these contractile cells. Oh, and there's the pretty picture that shows that. We did all of that. But what happens with the autorhythmic cells? The conduction cells or the autorhythmic cells are the ones that produce the main rate of the contraction of the heart. Let's take a look at this. We just finished talking about the contractile cells. We are now going to discuss the conductive cells, or as I also mentioned, what are known as the autorhythmic cells. Of the heart. Let's say it this way, cardiac conductive cells, let's say it that way. Now, one of the biggest differences in these conductive cells versus the cells we've been talking about so far is conductive cells do not have a resting membrane potential. So what that means is their membrane potential is always changing. But this constant changing is what allows them to produce their own action potentials. And that's taking up too much space. I need to make that smaller. So let's talk about how these cells work. Let's start first over here by drawing one of these cells. And then over here, we'll draw our graph again. Now, notice with our graph, we don't have a resting membrane potential. So we can't have a line for that. But let's talk about one of these things that are, we'll use purple. These conductive cells have some voltage gated channels. And what do voltage gated channels need to open them up?
Threshold.
Need a threshold, absolutely. So we may not have a resting membrane potential, but what we definitely have is a threshold. So let's draw a threshold, and we'll use brown. And we'll put the threshold right there. And we'll label it. That is our threshold. All right. Now, we can start our cell anywhere along this line because, again, we know its membrane potential is constantly changing. But our threshold is an easy place to start. So let's start there. When we reach threshold, we know that voltage gated channels are going to open. And a massive number of these channels are going to open. And we're going to have a massive influx of positive ions. And we're going to have a big, fast, rapid depolarization. And of course, we know a big, fast depolarization is our action potential. And of course, what ion is going to be responsible for this? Calcium. Isn't
Sodium.
sodium the one that does all of it? Sodium is what we are used to, but Ariel is actually 100% correct. What is actually opening here are voltage gated calcium channels. And we get a massive influx of calcium. Every other action potential we have ever talked about in this class has used sodium. But here, these are voltage gated calcium channels. Oh, I guess that needs to be purple. Now, as we know, cells don't like having calcium inside of them. So one of these things these cells always have are things like calcium pumps. These calcium pumps are, of course, going to use ATP. Oops, let's make our ATP yellow because it amuses me to do so. Uses that ATP to pump calcium out of the cell. Oops, that can be bigger. But with all this calcium rushing in these voltage gated calcium channels, are those poor little pumps going to be able to keep up with all of the calcium that is rushing in?
No.
No, absolutely not. So what's going to happen is we are going to get this massive influx of calcium. And this massive influx of calcium is going to cause a big, rapid depolarization. Which is an action potential. All right. Questions on that? Excellent. That's something new. Let's talk about something else new. Now, obviously, I put all of the calcium channels on one side. Are they just on one side of the cell? No, they're spread around the cell. I'm just doing this for a simple schematic. And I'll do the same thing with this second one as well. I'm going to put a bunch of other channels on the other side. Now, again, all of these channels aren't on one side and all of the others aren't on the other side, but this is a simple illustration that, again, illustrates our point. Make that one a little bigger, make that one a little bigger. This one's too big. There. The other type of special channel that this autorhythmic cell has on it is a calcium gated potassium channel. Let's unpack that. What does that mean?
The gate needs calcium in order to work.
Exactly. Calcium is the key that unlocks the door, but what is the ion that moves through the channel?
Potassium.
Potassium. Absolutely. Potassium is the ion that flows. So notice when I get this big influx of calcium into the cell, all this calcium is now inside of the cell. This calcium can bind to these calcium gated potassium channels. And when our calcium binds with the calcium gated potassium channels, what happens?
It opens them so the potassium can come in.
Is potassium going to come in?
No, it's gonna rush out.
Potassium is going to rush out. Absolutely. So when these open up, we get a massive efflux of potassium. There's lots of calcium. So we open lots of channels open. And we get a massive efflux of potassium. And if a massive amount of potassium leaves the cell, what happens to the cell?
Polarization.
We get a massive depolarization. So we had first a rapid depolarization. And that rapid depolarization, as we talked about, is due to calcium coming into the cell. But then we have a rapid, again, remember, we are calling this a rapid repolarization. But remember, repolarization means going back to the resting membrane potential. Remember, this cell doesn't have a resting membrane potential. So it's not really a true repolarization, but the cell is getting negative very quickly. So we use the word repolarization here, even though it's not 100% accurate. And again, this is occurring because of the rapid efflux of potassium. Positive ion leaves the cell, makes the cell more negative. But notice something interesting happened along the way. Oh, I'm going to cheat a little bit here. I'm going to grab that and move that like that. But notice something interesting happened along the way. What was that interesting thing that happened during this rapid repolarization?
Past threshold.
Yeah, notice on the way down, we passed threshold. And what happens when we pass threshold?
The voltage gated calcium channels will close.
And when those voltage gated calcium channels close, there is no new calcium entering the cell. However, there is still lots of calcium inside the cell. Can we do anything about that?
The
Pump them out.
calcium.
Yeah. Those pumps weren't very effective when the calcium channels were open, but now that the voltage gated calcium channels are closed, those pumps can remove the calcium. Now, are they going to be able to remove all the calcium instantaneously?
No.
No, this is going to take time. So what's going to happen is we're going to get a gradual decrease in calcium. So as calcium is being pumped out, and I will cheat and use some X's here. So that one gets pumped out, and this one, and this one, and this one, and this one. As calcium is getting pumped out, what happens to the number of our calcium gated potassium channels that are open?
They'll close.
Yeah, there's less calcium to open the calcium gated potassium channels. So our potassium channels start to close. And let's take it one step forward. Right now I have 4 channels that are open, allowing 4 potassium to leave. If I close one of them, then now only 3 potassium leaves, meaning more potassium stays inside the cell. And if more potassium stays inside the cell, what does that do to the cell?
Makes it more positive.
Yeah. And so what happens as a result of this is we get a slow depolarization. These potassium channels are slowly closing. More and more potassium stays inside the cell. The cell gets more and more positive, and we get this slow depolarization until what critical thing happens?
Reach threshold.
What happens when we reach threshold?
Voltage gated calcium channels open.
And we get a huge, massive influx of calcium, and we get a big, fast, rapid depolarization, and the process begins again. Wow, that was bad. Trying to line it up and make it look the same as the first one. So notice here with our conductive cell, with this autorhythmic cell, there are a couple very, very important things. The first is that there is no resting membrane potential. Notice this membrane potential never stays the same. It is constantly changing. We're constantly having the different flow of ions into and out of the cell. And so the membrane potential is constantly changing. And notice with that, there are basically 3 main stages to this process. We have the rapid depolarization. We have the rapid repolarization. And then we have the slow depolarization. And all of these 3 phases are driven by something different. Rapid depolarization is the amount of calcium coming into the cell. Rapid repolarization is by the massive amount of potassium that is exiting the cell. And our slow depolarization is due to the pumping out of calcium along with the closing, or we can think of it the decrease in the permeability of potassium. So less potassium leaves. And with less potassium leaving, the cell gets more positive. And that is what this illustration shows. Notice, oops, wrong button. Here, they started it already under threshold. So notice we are getting a slow depolarization, and that slow depolarization is occurring because potassium channels are closing. Don't worry about sodium. We'll talk about sodium in a second. Ignore sodium for right now. Potassium channels are closing, more potassium is inside. Cell gets more positive until we reach threshold. At threshold, those voltage gated calcium channels open and we get a massive influx of calcium. And that is our action potential, the big rapid depolarization. Ervek, did you have a question?
Yes. So can we say that potassium causes threshold, so it causes calcium channels to open, and calcium causes potassium channels to open?
Yeah, exactly. And that is the cyclical nature of this. Notice this process is a cycle. It is a continuous process. Notice that's why I was able to start at threshold. Your textbook starts at a different point because it is a continuous cycle. This is a continuous cycle that continues around and around and around. Absolutely. And so it is this relationship between calcium and potassium that keep this cycle going. Because when all of that calcium rushes in, as we mentioned, it's going to open all of those potassium channels, massive efflux of potassium. But when we close threshold, calcium levels go down. Calcium stops coming, calcium channels close, pump out the calcium, less calcium opening the channel, slow depolarization, and it continues.
And how does potassium enter the cell? Is it by sodium potassium pump, or there's a different?
Yeah, so again, notice we are focusing just on the two channels that are responsible for maintaining the cycle. But remember, as we've talked about many times, cells have hundreds of different types of channels. It has pumps like the sodium potassium pumps. It has leak channels and all these other channels. So calcium gated potassium channels are not the only way potassium can move. Potassium's constantly being pumped in, constantly finding ways to get out. So it's the changes in it. There may be 100 ways that potassium can get out, but if we start closing some of those potassium doors, then instead of 100 potassium leaving, only 85 are leaving. And so now as a result of that, more potassium stays inside. We're still pumping the same amount in, and it slowly gets more positive. You're absolutely correct.
So what I wanted to clarify, during the threshold, the only channel, even though there can be more channels, the only one that opens is the calcium channels.
Yeah, yes. At this threshold, the only channel that this cell has that is voltage gated is the voltage gated calcium channel.
Okay,
this is the only voltage gated channel that it has on it. And so that's why when we reach threshold, that's the only one we have to worry about.
Okay, thank you.
Yep. All right, questions on this? Again, I will ask the question strongly. Make sure this makes sense because it's about to get worse.
Yeah, I had a question.
Yeah.
Say this was an essay question. Where would we start exactly if there's no resting membrane potential?
That is the beauty of this. I don't care where you start. If you want to start at the top of the action potential, if you want to start at its lowest membrane potential, if you want to do like I did and start at threshold, because it's a cycle, it doesn't matter where you start as long as you go through all of the steps in the right order and show that it continues. So yeah, with a cyclic pattern like this, there truly is no starting point. So start anywhere on the wheel you want, just make sure you go all the way around the wheel. Great question. Any others?
I know this doesn't have to do with this per se, but is it the calcium that allows it to be able to beat on its own?
I'm not sure I'm understanding your question. You have, if you
Like
notice,
you always say the calcium makes cells do wonky things. Is this kind of like what one of the things that calcium does is it allows it to
Sure.
create its own
Look,
action potential?
absolutely. If you've noticed, Every other action potential we've talked about in this class has involved sodium. So the fact that this one does calcium is definitely something that is indeed wonky. And the fact that it does this on its own and doesn't have a resting memory potential is definitely something that's wonky. So yeah, I guess it's not surprising that calcium is involved in this very unique cell. All right. Like I said, now's the time to ask questions because this is about to get worse. All right, you're all quiet, so we will continue. So let's take this to the next step. As we have talked about, what's important and special about this is, like I said, this cell does this all on its own. As I mentioned, if I take this conductive cell and I put it in a petri dish and I leave it alone, it will cycle like this, producing action potentials. And as I mentioned, it produces those action potentials at a rate of about 100 per minute. Which is great, except for the fact, do you want your heart beating 100 beats a minute all the time?
No.
No.
No. So while it does this all on its own, we need to be able to modify it. And how did we say we would modify it?
Sodium.
Well, not specific. Well, how do we modify your heart rate?
Parasympathetic.
Yeah, there you go. Yeah, well, not just parasympathetic, but the entire autonomic nervous system. And as I heard, hormones, right? What does the autonomic nervous system release to its target cells?
Neurotransmitters.
Yeah, I'll just put NTs here. Neurotransmitters. And hormones, which are a chemical signal. So notice the way we're going to modify this is with chemical signals. And lucky for us, chemical signals are really good at opening things like chemically gated channels. So let's put one of those here. Let's put a chemically gated channel. And let's say, for instance, this chemically gated channel happens to be gated by, oh, I don't know. Let's say this one's associated with the sympathetic nervous system. What would the chemical be released by the sympathetic nervous system that would modify the activity of the heart?
Adrenaline.
Excellent. Adrenaline or epinephrine would definitely be something that our adrenal gland would release into the blood. Anything else? What's the actual neurotransmitter that our sympathetic nervous system uses? It's not epinephrine, but it was something very similar to that. There it is, norepinephrine. Ariel's got it. Norepinephrine. Excellent. So an epinephrine or norepinephrine, and remember for our purposes, they basically do the exact same thing. So we have an epinephrine or a norepinephrine that binds to this channel. And does anybody remember what our sympathetic nervous system, what kind of channels it opens on the heart with that epinephrine or that norepinephrine? All right, let's work our way to it. What effect is the sympathetic nervous system going to have on our heart?
Excitatory.
Makes it beat faster. So we want to excite it. And if we want to excite it, what type of chemically gated channel, what ion do we typically want to let flow if we want to excite a cell?
Calcium.
Well, true, but remember, we're already letting calcium in. What's the one that we normally use to depolarize a cell, to
Sodium.
excite a cell? Sodium. So here we have our starting point again, and let's start here with our starting point with a nice big circle for our starting point. But now we're going to affect this conductive cell with our epinephrine, with our norepinephrine, opening sodium channels. Now, is sodium coming in really going to affect threshold? Does it change when the threshold is? Not a trick question. Is it going to affect how much calcium comes in?
No.
No. So notice our rapid depolarization is going to be the same. And we're going to get the same amount of calcium coming in, which means we're going to allow the same amount of potassium to leave. So notice as a result of that, my rapid repolarization stays the same as well. But now, here with my slow depolarization, not only do I have, like I said, more potassium staying inside the cell, but now I'm also letting sodium into the cell. By opening up these these sodium channels, I'm letting sodium into the cell. So actually, let's just start there. I'm letting sodium into the cell. And if I'm letting sodium into the cell, what effect is that going to have on the cell?
Making it more positive.
Excellent. It depolarizes the cell. And if it depolarizes the cell, am I going to reach threshold faster, slower, or at the same rate that I would have before?
Faster.
So now what's happening is I am depolarizing it much faster, and I reach threshold much faster. And what happens when I reach threshold?
Voltage gated calcium channels open, massive influx of calcium.
There you go. Massive influx of calcium, massive efflux of potassium. And then I start a rapid depolarization again. And notice by using my sympathetic nervous system, by opening these sodium channels, by increasing the speed of the slow depolarization, what have I done to my heart rate?
Sped it up.
Yeah. As a result, I get a faster heart rate. I make a point of emphasizing this because this is not a distinction I think your book does a good job of doing. Right? I absolutely, again, sodium does not play a role in the normal rate of the heart. Sodium's primary role is to modify the heart rate, make it beat faster.
Professor?
Yes.
For the parasympathetic nervous system, what channels would you have then?
I think that's an excellent question that we should figure out the answer to. But let's make sure we understand this one first before we go to that one. So that is exactly where we're going next. I love that question. Absolutely, that's what we're going to do next. But I want to make sure we understand this before we move forward. So any other questions on what's happening with sympathetic nervous system or excitatory hormones and how we make the heart beat faster? All right, excellent. Then let's answer that perfectly spectacular question. And we will do that by putting orange channel. So this would be, for instance, our parasympathetic nervous system or some type of inhibitory hormone. What is the neurotransmitter that is used by the postganglionic neurons in the parasympathetic pathway?
Acetylcholine.
There we go. Excellent. Acetylcholine is the hormone. So we're going to have some acetylcholine gated channels. And does anybody remember what type of channels these are? We talked about it in 430. We briefly mentioned it, so it's okay if you don't remember it. But does anybody remember what kind of channels acetylcholine opens on the heart muscle?
Chemically gated?
True, they are chemically gated, but the ion that flows through them is actually chloride. So when we open chloride channels, which way does chloride ions move?
In the cell?
And chloride are negatively charged ions. So when these negatively charged ions enter the cell, what's the effect?
Makes the cell more negative.
Yeah. So notice, once again, we have our starting point. We're going to get that rapid depolarization. We're going to get that rapid repolarization. But now we're opening chloride channels. So yes, potassium is not leaving the cell, making the cell more positive, but also chloride ions are entering the cell. And so if those chloride ions are entering the cell, making the cell more negative, what's the effect?
Longer depolarization, or sorry, repolarization.
Right,
Further
absolutely.
down the graph.
Exactly. It's going to take longer to reach threshold. So now it's going to take a longer period of time before it reaches threshold. And if it takes a longer period of time to reach threshold, what effect does that have on the heart rate?
Slows down.
And there you go. Notice what's so awesome about this and how elegant the system is. Not only have we learned how these conductive cells are capable of producing their own action potentials and setting the basic rate for the heart, but we now took that information that we learned in 430 about how the sympathetic nervous system makes the heartbeat faster and the parasympathetic nervous system makes the heartbeat slower. And now we've actually seen how and why that occurs.
I had a question.
Yes.
For both the parasympathetic and sympathetic nervous system, at the lowest point of the membrane potential, what causes the chloride channels and the sodium channels to open?
So when they're opening all the time, but the amount of them that are coming in during the first two phases are not as great as the ions, as the calcium and the potassium that are flowing. So you don't see as much of an effect. However, you do see some effect because obviously when your heart beats faster, notice it also beats stronger as well. So you do see a little bit of an enhancement of the contraction and a little bit more strength in that. Again, remember, what we're doing here is the sky is blue version of this. All of this, believe it or not, is far more complicated than we're even talking about it right here. So yes, there are some layers to it. There are some implications to those, to the strength of the contractions as well, and some other factors along that way as well. But at its core, this is the basic component. This is the sky is blue version of this.
Thank you.
Yep. All right. Questions now on this. Amanda, did you try to say something?
I was going to ask about like exercise and heart rate increase. Would this be the same way or is that something different?
No, it's exactly the same way, right? As we talked about, when you're physically active, that is stimulating your sympathetic nervous system. Your sympathetic nervous system is releasing epinephrine, releasing norepinephrine, and that is speeding up the heart rate, making the heartbeat stronger to pump blood to more parts of the body to meet those increased demands. Absolutely. This is exactly how the sympathetic nervous system would do that for something like exercise.
Thank you.
Yep. All righty. Here's what we're going to do. This is a good stopping point for our second break. Let's go ahead and take this. Although before I take our break, there's one thing I want to point out and emphasize first. I've said it earlier and I will say it again in this class. People often lose points, not because they don't know the information, but because they don't read the questions carefully. We just spent a whole hour talking about cardiac conductive cells and talking about cardiac contractile cells. Two very important and very similar cells. As such, how many possible essay questions could you have from something like that?
Three.
At least three, and in this case, probably four good ones, but give me the three.
Explain the contractile cycle of cardiac muscle cells, explain the twitch or the conductive, the cycle for conductive cells, and compare the two. Absolutely.
So you're absolutely right. Notice both the contractile cell and the conductive cell have action potentials. So describe the action potential of the contractile cell, describe the action potential of the conductive cell, compare the two action potentials. And this slide here could probably be an essay question as well. Compare the contractile cardiac muscle cells, action potentials and twitches to the action potentials and twitches of skeletal muscle. So really for this part, we've had four, right? But the two I want to talk about right now are contractile cardiac muscle cell action potentials and conductive cardiac muscle cell action potentials. Because despite all my warnings on every single test, there's at least one student who, when I ask for the muscle action potential of the cardiac contractile cell, gives me this. Beautiful information, 100% accurate and correct. They even do beautiful drawings and all of those things. And the problem with that is while every single piece of that information is correct, I asked for the contractile cell. So if I ask for the contractile cell, don't give me the conductive cell. If I ask for the conductive cell, don't give me the contractile cell. People often lose points, not because they don't know the information, but because they don't read the questions carefully. Read the questions carefully so you can answer them correctly. All right.
Professor?
Yes.
I have a question. It relates more to lab though.
Sure.
So for the blood vessels, I started studying those, but I was having a little bit of trouble kind of grounding myself because some of the pictures, like some of them are diagrams of like you see the full body, and then some of them are just diagrams that you just see bone and then you see the blood vessels. Do you have any tips for studying that, or maybe should I wait till we go over it a bit more in lecture?
Well, like I said, we will it might not be till Tuesday the 7th that we get a chance to go over it in lecture. And that's actually after the exercise is due. So I wouldn't necessarily just rely on that. Again, it's one of those challenging questions to answer because there's no one right way to study this material. What works best for you. For some people, I think they do better starting with the full body so that they can better relate themselves that way. It's kind of like if you were going to a new city, right? You want to get the main streets, right? You need to know where Greenback is. You need to know where Madison is. You need to know where Auburn is, right? You need to get those big ones done. And as you get familiar with that, then you can look more closely at, all right, what are all of the side streets that come off of Madison? What are all the side streets that come off of Auburn Boulevard? So I think for some people, if you're getting a little overwhelmed by the information, starting big with the big picture and getting more comfortable with the general organization, and then focusing on those up close pictures where you just see an arm or just see a leg. And that might be a way to ground yourself a little bit more if you're having that trouble.
Got it. Thank you.
Yep. The other thing I would say is there's lots of different modalities for this too. There are the pictures in the textbook. There are the good models that we have in the on the PAL CD. So again, a lot of it also has to do with your learning style. If you're a very tactile learner, then there are some great models. We actually have some blood vessel trees basically at the open lab. So if you're able to make it to the open lab, if you're a very tactile learner, that could be something that could be helpful as well. And so maybe being able to actually better see that three-dimensional relationship can be something that can be very helpful as well. All right. Any other questions on this? I think I just dumped a lot of information at you, so I think we are good for another break. So any questions on this before we take our next break? All right, I think I saved this image, but I will do it again just to make sure. All right, let's go ahead and take our second break. Take another 15-minute break. Looks like it is 2:38, so that means we will restart at, what is that going to be, 2:52, 2:53? And I will start the recording at that time. All righty, questions on any of that? All right, I will see you guys in 15 minutes. We are back. We have now talked about both types of cardiac muscle cells, the contractile cells and the conduction cells, or what are known as the autorhythmic cells. And we have seen their action potentials as well. So I'll ask one more time now that you've had a break to decompress and think. Are there any more questions about this information as we move forward? Excellent. Stunned silences are the things I love the most because that tells me that you guys have mastered everything and I get to make the tests harder. So that is awesome. Excellent. So let's move on to our next concept. Now, these conductive cells sound pretty darn important. Do you think they're just randomly distributed throughout the heart?
No.
No, of course not. There is a very precise arrangement to how these are aligned in the heart. And as such, not surprisingly, the conductive cells are organized into a structure we call the cardiac conduction system. There are 5 main parts to this cardiac conduction system. Now, it is important to understand that all of these parts of the cardiac conduction system all do not have a resting membrane potential, all are autorhythmic, and therefore produce their own action potentials, fire spontaneously. But they don't all fire at the same rate. Some of them are faster than others. For instance, the fastest is a structure known as the sinoatrial node. And that has a beat of, like I said, about 90 to 100 beats per minute. There is a second node named the atrioventricular node. It, on the other hand, beats at a rate of about 40 to 50 beats per minute. Could you survive with that, a heart rate of 50 beats per minute?
Maybe if you're an athlete.
True, you are correct. Some athletes do have a decreased heart rate because their heart becomes more efficient at pumping. But do you think you could actually be an athlete if your normal rate of your heart was just 50 beats a minute?
No.
No, probably not. You would survive. You could absolutely survive that, but going up a flight of stairs or two might make you especially winded, right? A long walk, you certainly would have trouble running or riding a bike or swimming or things along those lines, but you could survive. And there are other additional fibers in the ventricles that have a spontaneous rate of low as 20 to 40 beats per minute. That's probably not enough to keep you alive, but they have this spontaneous rate. So the spontaneous rate can vary pretty dramatically between the regions of these. Now, not surprisingly, it is the sinoatrial node, which after you've written that out once on an essay question, you can abbreviate it the SA node, that normally sets our heart rate. And so why do you think it's the one that normally sets the heart rate?
Because it beats the most.
Yeah, it's the fastest. Basically, all of these cells are sitting here interconnected to each other, right below threshold. And then the SA node cell is the one that fires first. And when it fires its action potential, it spreads to all the other ones and it goes through the system. And then they all reset. And again, because that SA node is the one that is the fastest, it fires its action potential first and it spreads through all the other ones. Right? If you ripped out that SA node, other cells would be able to fire and drive the process, but they fire at a much slower rate. So the SA node has the highest spontaneous rate. And so because it has the highest spontaneous rate, it sets the autorhythmic rate of the heart. But as we've already talked about, your heart rate can be modified. We've actually seen how it can be sped up and how it can be slowed down. And what were those things that modified it?
The sympathetic and parasympathetic.
Sympathetic, parasympathetic.
Yeah, sympathetic nervous system, parasympathetic nervous system, and
Hormones.
hormones. There you go. Absolutely. Now, if I had a heart rate of 135 beats per minute, would I be tachycardic?
Yes.
All right, I got one yes, I got one no. Anybody else have an opinion on it? Technically, I got two yeses. I'm sorry, go ahead, Arabic.
Depends for how long.
Okay, part of it depends on how long. It also depends on the activity. There you go, absolutely. If I am running in a triathlon, would it be appropriate for me to have a rate of 130 beats per minute? Yeah, absolutely. If I'm relaxing in a bathtub, should I have a heart rate of 130 beats per minute?
No.
No. And that's kind of the point I want to make. Obviously, when we talk about irregularities of a heart rate, tachycardia is a higher than expected rate. And of course, bradycardia is what?
Lower.
Slower.
Lower than expected. But you can't just limit that to a number. It's really higher than or lower than expected based on the activity. The bear with an axe walks in this room and I'm fighting that bear with an axe and my heart rate is beating at 50 beats per minute, right? That is very low for that situation. However, if I'm in my yoga class or meditating or something along those lines, then sure, especially if I'm doing a deep meditation, a heart rate of 50 is not, outrageous. It's a little low, but still. So again, when you think about these irregularities, make sure it is just not a number, but it is associated with the appropriateness of an activity. There are also lots of things that can be what we call ectopic pacemakers, medicines, caffeine, other types of recreational drugs, things along those lines have the ability to modify our heart rate artificially. And then, of course, if you have a heart condition, they actually may put in a pacemaker, an electrical device that produces its own action potentials to drive the heart rate of the heart and maintain the normal pacing of it. So they can both be chemically and they can also be electrically induced. So like I said, caffeine, nicotine is another great example. And then, like I said, obviously artificial pacemakers stimulate that as well. But let's not talk about these abnormalities. Let's focus more on the normal healthy heart. Here we see a normal healthy heart, and we see our illustration of the cardiac conduction system and its 5 components. Now, this is a very nice illustration that does an excellent job of showing this, but as you all know, I am an amazing artist as well, so it's useful for me to take some time to draw this as well. So let's do that. Of course, first I have to draw my absolutely accurate, oh, no, not thick enough, representation of the heart. Excellent. And then we have to distinguish the chambers. Excellent. So there you go. Here is my amazing recreation of the heart, which is going to allow us to identify the components of the cardiac conduction system. As I mentioned, the first part of the cardiac conduction system is the sinoatrial node, which, like I said, after you've written it out once, you can abbreviate it as the SA node. Oops. It is basically found inside of the right atrium, kind of in the armpit of the superior vena cava. Where the superior vena cava enters into the heart, kind of in the armpit of that right there, is where you find the sinoatrial node. As we mentioned, this is what is commonly referred to as the pacemaker, because its job is to start the heart rate. Now, when it activates and the cells generate that action potential is then going to spread and then spreads that action potential to all the contractile cells that are around it via our gap junctions. And then, of course, those contractile cells are going to spread it through their gap junctions to the cells next to them, and they're going to spread it to the cells next to them. And so basically what happens is that this action potential is spread throughout the atrium. This leads to the depolarization of the atrium. And again, that makes sense. If you stand at the end of the pool and throw a big rock into the pool, it produces a wave, and that wave spreads. And that's exactly what happens here. However, our wave in the pool spreads over the entire pool. And that's not the case here. This depolarization only spreads across the atrium. It does not reach the ventricles. Well, why not? Well, for the same reason, when you drop that rock into your pool, it produces a big wave in the pool, but does it produce a wave in your hot tub?
No.
No, right? Because typically there's a wall between your pool and your hot tub. And it turns out there's a wall here as well. Remember, we talked about how there is that fibrous skeleton. And we talked about how that fibrous skeleton is thicker in the area where the valves are. And so basically that fibrous skeleton stops the spread of the action potential from traveling from the atria to the ventricles. Now, don't we need that action potential to travel from the atria to the ventricles? Yeah, absolutely. But lucky for us, there is a second structure in our cardiac conduction system. And that second structure is the atrioventricular node. Oops. Which again, after you've written it out once on an exam, you can, essay question, you can abbreviate it the AV node. And guess where the AV node might be located?
In the right atrium?
Right atrium.
Well, true, but just in the right atrium? It's the atrioventricular node after all.
It connects the right atrium and the right ventricle?
Close. It is within the wall, within the muscular septum between the atria and the ventricles. So it's in the septum. That's way too big. It's in the septum between the atria and the ventricles. So its location of this AV node is important. Oh, yeah. But there's also something else very important about this AV node. The cells of the AV node are very small and thin. Why might that be significant? Because it gets bullied and needs more counseling.
Is it for permeability?
I'm sorry, Amanda, your microphone is really hard to understand. I greatly appreciate that you're trying to be active, but it's really hard to understand you. I do apologize for that. Can you type it?
Easily impressed.
I like that. Permeability is a great way of describing it. Absolutely. Right. Remember, when we're talking about action potentials, we're talking about the flow of ions. We're talking about flow, right? And from a physics standpoint, right, the enemy of flow is resistance, right? Again, you may not have thought of it in these terms, but you're aware of it because when you go to McDonald's and buy a shake, do you ask them for a coffee stirrer that you can use to drink that shake? Or do they give you one of those big nickel-sized McDonald's straws that you can drink that shake through?
Big one.
Right. Because notice the less in contact with the wall of that straw the fluid is, the easier it is for that fluid to flow. So if these cells are very small and very thin, what happens to their resistance? Is it going to be very high or very low resistance?
Low.
The resistance is
Very
going to
high.
be low. The resistance is going to be high. And with that high resistance, oops, that means it is going to slow the movement of the action potential down. It adds a delay. Why might that delay be important?
So it doesn't contract at the same time.
Right, absolutely. Remember, this action potential that's spreading across the atria is stimulating the atria. They're producing action potentials. And as we know, when those contractile cells produce an action potential, they're going to contract. Now, do they contract instantaneously the second they get stimulated?
No.
No, we know there's that latent period. There's a bit of a delay before it starts producing its tension. And so we don't want the atria and the ventricles contracting at the same time. That would not be an efficient pumping motion. So we want to give the atria a chance to finish pumping its blood into the ventricles before the ventricles contract. So this delay plays a very important role in helping to organize and coordinate the pumping of the heart. Now, coming out of this AV node is a structure, a singular structure known as the bundle of His. Why is it called the bundle of His? Come on, people who had me for 430 know why.
Because he was the one discovering.
Exactly. Good old Bob Hiss was an anatomist who discovered this, took a picture with it, drew it, something like that, published it in a textbook and named it after himself. Absolutely. It's also called the atrioventricular bundle because it's the bundle that comes out of the atrioventricular node. And both of those are acceptable terms for it. And its job is basically to, I forgot to put my 2 there so that I can put my 3 here. For that AV bundle. The job of the AV bundle is basically to transfer our electrical signal from the atria to the ventricles. It is a singular structure that does this. The AV node and the bundle of His, these are the 2 structures that when people have heart blocks, these are the, where there's a blockage in the flow of the electrical signal of the normal flow of the cardiac conduction system. These are the 2 areas where they most commonly occur because this is the narrowest part of this path, right? If just one of these paths gets blocked here between 2 contractile cells, there's dozens of other gap junctions that it can move around to get through. But if this one singular bundle gets blocked or damaged, then that can cause some serious problems. In fact, your interactive physiology will deal with that a little bit when you're doing that homework assignment. Excellent. That one singular bundle of His, that bundle, atrioventricular bundle, then branches, no, wrong button, branches to form numerous structures known as the bundle branches. And the bundle branches, you will notice, bring the action potential to the apex of the heart. Once it reaches the apex of the heart, then there are additional branches that take it up the walls of the ventricles. And those additional branches that take it up the wall of the ventricles are known as the Purkinje fibers. Some of you may have remembered the Purkinje cells that we found in the cerebellum way back in 430. Well, this is the same good old Bob Purkinje who discovered these in the heart and, of course, named them after himself as well. Like I said, there are like something like 5 different things in the body named after Purkinje. He was what we affectionately like to refer to as an anatomy whore. Excellent. Two more things I want to add to this. The first is that while these signals are traveling down the interventricular septum, the big muscular wall that is down the center of our heart between the two ventricles, there are some small offshoots off of this that will actually go to those papillary muscles. Remember how we talked about those papillary muscles have the chordae tendinea. Oh, here, let's draw one on this side so it's not in the way. That connect to the cusps of our valves. And remember, we talked about how they have to produce that tension to stop the valve from prolapsing. Well, these help to allow that to occur. So again, remember, we want them contracting a little bit sooner before the ventricle is, so that tension is in place. In fact, there's one other thing I want to mention as well. 100% true in sheep may or may not be true in humans. If you remember, we talked about how the left side of the ventricle, the left ventricle, has all the advantages. It has a circular shape, it has a thicker muscular wall to produce a much more powerful contraction, whereas the right side just kind of weakly wraps around it. Thinner muscle wall, less efficient shape, and doesn't contract as well. So definitely in sheep and maybe in humans, there is an extension of the wall of the, a tube-like extension of muscle that goes from the interventricular septum to the wall of the right ventricle, in which one of these or some of these bundle branches can take an alternate path to get here to the ventricles and start the contraction a little bit early to give it a little bit of help. Anybody know what this particular interesting, unique structure might be called?
Moderator band?
Exactly. As you look at different heart models, one of the things you'll notice is some of them have a moderator band in them and some of them don't. And the reason for that is anatomists can't come to a consensus as to whether or not humans have them. It appears some human hearts look like they have them, some don't, but it's hard to tell. And obviously it's not something we can watch functioning inside of a human heart while it's working. So it's still a debate. But what's not debated is that it is always found in a sheep's heart. So the easiest place to see a moderator band is actually to look at it as a sheep's heart. And as I mentioned, if we were in class, we'd actually be doing a sheep heart dissection today, and you would get a chance to see that. But unfortunately, we are not.
Professor?
Yes.
Sorry, so just to clarify, so the moderator band is kind of like a tunnel for the bundle branches to get to the wall of the right ventricle, just like a different way?
Correct.
Okay.
Yeah, to get, it's kind of a shortcut that the electrical signal can take to try to help to make the contraction of the right ventricle more efficient.
Thank you.
Yeah, and actually, you know, I don't actually know. Let's take a quick peek at the picture from your textbook to see. Yeah, see, notice even the picture of your textbook for the cardiac conduction system doesn't show us a moderator band. So like I said, there's no consensus as to whether or not it is there in humans, but it's definitely there in sheep. Excellent. All right, that is all of my questions except for one. And that one question is why? Why not just let the electrical signal spread across the entire heart? Why do we have to bring the action potential to the AV node, bring it through the bundle, bring it all the way down to the apex before we finally bring it up the sides and cause the contraction. Why is it built this way?
Wouldn't that completely change how the heart works and the motion in and of itself?
True, but so why did it arrange itself this way? Wouldn't it be easier to just have it spread like a pool all the way across the surface? We wouldn't have to have this elaborate node or this elaborate bundle.
That's not very efficient, I don't think.
True. So part of it is the efficiency of the delay. Definitely when it comes to why, and again, remember, why is always one of these questions that's hard to answer in anatomy and physiology, because of course, the engineer who designed this did not leave us her footnotes. But from a mechanical standpoint, we can see two huge advantages. One of them absolutely is the delay that we talked about that is going to give you more of a sequential contraction of the heart. And we already talked about that with the AV node. But as you're also talking about with an efficiency standpoint, the other thing that this does is it gives a directionality to the contraction. How do you squeeze a tube of toothpaste? Okay, hold on. Let me rephrase that. How are you supposed to squeeze a tube of toothpaste?
From the bottom.
From the bottom, because when you squeeze it from the bottom, you get this very efficient pumping action. Now, like I said, nobody actually does that. Everybody squeezes it in the middle, but you're supposed to squeeze it from the bottom, and that is the most efficient way to move the toothpaste out. Well, remember, I've got a very simple schematic of my heart here, but remember, the great arteries are located at the top of the heart. So by bringing the electrical signal all the way down to the bottom and then having it travel up the walls of the ventricles gives us a nice directional contraction of the ventricles that gives us an incredibly efficient pumping action. And while my illustration doesn't do a good job of showing this, if we look at a real heart, and again, here they've done a nice job of showing us the left ventricle. Notice by bringing the signal down, we can get this directional pump that will pump the blood up and out into the aorta. And this one will pump it up and out, even though it's not drawn here, into the pulmonary trunk. So it gives us this nice directionality. Right. Rolling up the toothpaste from the bottom gives that same directionality to it. Absolutely. All right. We've done a good job of drawing this with the schematic, but like I said, we've got these pretty pictures from your textbook that do a nice job of showing this as well. Again, our sinoatrial node are the cells that are most rapidly spontaneously active. So they are our pacemakers. They're the ones that produce the original action potential or the starting action potential. That action potential spreads across all the contractile cells through all those gap junctions. And of course, what we get is three things that happen. We get a depolarization of the contractile cells. Of course, when the contractile cells depolarize, our contractile cells produce action potentials. And those action potentials continue to spread. But what's the other big event that occurs in these contractile cells when they produce an action potential? I know it's late. I know it's a lot of information, but these are the easy questions. What do contractile cells do when stimulated?
Contract.
Contract. And when these contractile cells contract, our atria pump the blood into the ventricles. There you go. So we get this spread of the action potential across the atria, stimulating the contractile cells, causing those contractile cells to contract, pumping the blood into the ventricles.
Professor,
Yes.
if someone has mitral valve prolapse, the reason for that could be, is it the dysfunction of AV node or a nerve?
No, it more has to do most likely, because again, remember, as we talked about, the left ventricle is the one that produces the most amount of pressure. And so as it's producing that pressure, it's pushing against the mitral valve and then being forced out in the semilunar valve into the aorta. But if it pushes on that valve, if the valve prolapses, then what happens essentially is this doesn't seal properly. So when it contracts, some of the blood is going to go back into the atria instead of going out into the artery. So it's really not, it's related more to the changes in pressure and damage to the valve as opposed to anything to do with the conduction system.
Okay.
Muscle in the right ventricle. You are correct in that definitely the ventricles have much thicker walls. Even the right ventricle has a thicker wall than the atria because they're having to pump the blood out into the arteries, where basically all the atria have to do is pump it into the ventricles. And as we'll see when we talk about the cardiac cycle, gravity does a lot of the atria's jobs as well. So yeah, absolutely. They're less muscular than the ventricles because they have to do much less work. Yeah, and that is probably due to the orientation. I'm not necessarily sure if that's a reason why it's tilted to the side and anteriorly. That I don't know how much that would change the efficiency, but definitely having the atria superior and the ventricles inferior is the main reason. The amount of pressure the ventricles have to produce is definitely why there's more wall, more muscle in the ventricle than the atria. Yeah.
So, Professor,
yeah,
is that the reason why astronauts have to go through extensive heart training to make sure that it's healthy and efficient enough because they don't have the assistance of gravity?
Sure, absolutely. That is definitely one of the reasons. Yes, absolutely. Also, the G-forces put tremendous strain on the body, so that's the other reason that they want. So the acceleration of going up is a major. I mean, the G-forces are over 5G often for that type of thing, and so it can be so very potentially damaging to the heart as well. Because pressure makes the world go round. All right, excellent. So we get that nice, efficient stimulation of the atria, which leads to that nice, efficient contraction of the atria. And as we talked about, we don't want the ventricles contracting at the same time. And so luckily, that's the goal of the atrioventricular node, right? The fibrous skeleton is thick enough in this region to disrupt the normal wave-like flow of the signal. So the only way that our action potential can get from the atria to the ventricle is through the AV node and the AV bundle. And the problem with the AV node, as we mentioned, is that they're very small and very thin cells, which dramatically increases their resistance, decreasing the flow, right, of that action potential, slowing that action potential down. Right? And as you can see here, about 100 millisecond delay. Eventually, it sneaks its way into the bundle of His and the bundle branches. So you notice that previous picture didn't, and again, notice this one, though, it does show a moderator band. So like I said, the models, illustrations, some artists will put them in there, some artists won't. The bundle of His and the bundle branches take the signal down the interventricular septum, the muscle wall between the two ventricles, to the apex of the heart. And from there, it travels up the Purkinje fibers. And again, the reason we bring it to the apex of the heart is for the directionality. So we can get that nice, smooth directional pumping of the ventricles to efficiently move the blood where we need it to go. And so from there, it spreads up the ventricles. Notice our atria are relaxed, or at least they're repolarized at that time. And the ventricles are going to contract. And as we talked about, those papillary muscles will contract slightly before to maintain the valves and hopefully stop them from prolapsing. All right. So there you go. Those are the 5 components of the cardiac conduction system and how they help to produce and spread that action potential through the heart, leading to the rhythmic and efficient contraction of the heart. Questions on that?
I have another question, Professor. I don't know if I
Yes.
missed this part, but we've talked about the function of contractile and conductive cells, but do we have to know their location, how they're, do they communicate to each other, or how to differentiate them histologically?
Yes, yes, and yes. So all 3 of those are great questions, and the answer to all 3 of those are correct. No, I don't know what happened, what page number these actually are, but they're in your textbook, I know. Yes, obviously, like I said, the conductive cells only make up 1%, and they're not randomly distributed. They are found in these 5 specific locations. Now, as we also know, these are connected via gap junctions to the contractile cell because they're the ones that produce the action potential that then spreads to the contractile cells. Remember, contractile cells have a resting membrane potential. If you leave them alone, they'll stay at that resting membrane forever. So these cells are producing and spreading the action potential that is going to stimulate the muscle cells to contract. All right, that part makes sense?
Yes. Mm-hmm.
Excellent. So then let's answer your 3rd question. The 3rd question you asked is about identifying them histologically, and if that is possible. Here was the last slide we looked at when we looked at some histology on Tuesday. We saw the intercalated discs. We saw all the striations. And as we talked about, what do these striations indicate?
Sarcomeres?
Sarcomeres, myofibrils. And so of our 2 types of cardiac cells, which ones need sarcomeres? Which ones need myofibrils? Which ones need to be able to change their shape and produce tension?
Contractile.
Contractile cells. Notice all the cells we see here are indeed contractile cells. Conductive cells don't produce tension. They produce action potentials. They don't have myofibrils. They don't have transverse tubules. They don't have the sarcoplasmic reticulum in the same way. In many ways, the conductive cells are kind of a cross between a muscle cell and a nervous cell. And so not surprisingly, they look very different. The easiest place to see them is actually in the myocardium of the ventricles, because the ventricles have the biggest, thickest muscular wall. And what you're going to see in here are these cells that look dramatically different. They almost look a little bit like adipose, but not as clear as adipose. And remember also with adipose, the nucleus is always shoved to the side by that big oil droplet. Notice here, the nuclei are located in the middle of the cells. They're not shoved to the side in the same way. So we have these big kind of pillowy, non-striated cells that are not fat, not muscle tissue, clearly something else. And is that something else something you're able to distinguish histologically? Absolutely. And so if you notice actually on your histology slide, your histology handout, you are responsible for these Purkinje fibers. This one happens to be right at the edge, right next to the endocardium, the inside of the heart. You sometimes will see these clustered between the fascicles, between the bundles of it as well. So they don't always have to be on the edge, but they're always going to be in the myocardium, and they're always going to be separated from the myocardium, kind of forming in these cloud-like clusters in between them. And so they are typically something that is fairly easy to recognize histologically. So yes, to answer your third question, you are responsible for distinguishing contractile cells versus conductive cells histologically. And in fact, you stole my thunder, because if you cheat and we go back to the lecture, notice I had a reminder here to show you that histology and make sure that we saw those Purkinje cells. So thank you very much for asking that question, because that is exactly where I wanted to go next. Let's go back to that slide. Questions on that? All righty. You guys were not as interactive today. I will give you the benefit of the doubt because this is the first week. But again, as I said before, if I just wanted to sit here and listen to myself talk, I would record the lectures and I'd be sitting by the pool drinking pina coladas right now. So again, I appreciate and encourage enthusiastic interaction. This is dense, hard material. Ask questions so that we can get through it, because right now we're done. That is the information that I wanted to cover for today. I will leave us with one more thought as we move into the weekend and you start working on this on your own. As we, oh, I don't think I brought that up for me. I don't think I brought it up for you. So let's do this. As I bring this up for you, what we are talking about and seeing here is a movement of electrical activity traveling through the heart. And is it possible to measure the movement of electrical activity?
Yes.
Yeah.
Might it be useful to measure the movement of electrical activity of the heart?
Yes.
Yeah.
Absolutely. And so what kind of tool might we use for something like that?
ECG or EKG.
Exactly. An electrocardiogram or an electrocardiogram, CK, depending on whether or not you're German. Exactly. So that movement of the electrical activity can be measured, and we can learn a tremendous amount about the health of the heart by looking at these deflections, essentially, in the movements of the heart and understanding what they tell us. And that is what we are going to be doing on Tuesday. On Tuesday, we are going to be talking about this in class. Then you also have your interactive physiology 2.0 activity on the EKG that you will be doing and moving forward with that. All right. Excellent. Claudia, that is a great question. So let's actually talk a little bit about this, because actually I was dealing with that today, actually yesterday, in my office hours with my 430 students. It is perfectly okay and acceptable to feel overwhelmed right now. Think about this. In 3 days, you have had 9 hours of new information in 3 days, right? That's incredibly overwhelming. It's impossible to gain and retain that information. It's impossible to pay attention for that long. Heck, I'm not barely able to do it, and I'm the one saying all this stuff. It is okay to be a little overwhelmed right now. But what you have to remember is we don't meet again until Tuesday. So you have 5 days to process this information, go over this information again, and work with this information. The point of my lecture isn't so that you have completely mastered this information after the words have come out of my mouth. The goal of my lecture is to walk you through the information, introduce you to the information, so that as you are studying it and looking at it moving forward, you have me as that frame of reference to fall back onto to help you to understand and comprehend and ultimately master that information. All right. Now, you ask about how you can prepare for next week. That's a great way to do it. One of the things you can start doing is start preparing ahead of time. And the best way to know what to look at for next week is to look at the lecture slides, because the lecture slides are basically what I'm going to be talking about next week. Now, the one thing I do want to warn you is I am a bit of a tinkerer. Obviously, all the lecture slides have been posted, and I do tend to tinker with them when I'm in office hours and no one's meeting with me. I get bored and I look at the lectures. I may change the wording. I may switch a slide or two around or do things like that. So the lecture slides aren't gospel, but they are definitely a good outline of the important concepts we're going to be covering. So if you look at the lecture slides, you'll be able to see that on Tuesday, we're going to be talking about the ECG. We're going to be talking about the cardiac cycle, right? From there, we're going to go on to regulating the heart rate, controlling and regulation of heart rate, and all of those types of things. So it is a great guide to show you what to look at. So see that we're going to talk about the ECG, and then go to your textbook and read the part where it talks about the ECG, or go to your interactive physiology where it talks about ECG. Yes, you have an assignment on it, but even if you didn't, it'd be a good study tool. And there's lots of other resources that you can be doing as well. So yes, the next 5 days can be used to master all the information we've just covered, but you can also correctly use this time to start preparing to move ahead as well. So like I said, it is perfectly acceptable, perfectly expected to be a little overwhelmed at this moment in time. Because like I said, we've had 9 hours of new information in 3 days. But you have 5 days to process that information and make sense of it. If you're frustrated and confused now, that is okay. If Saturday, Sunday, or Monday, you're still confused and still frustrated, then that's when you need to meet with me, and we need to talk, and we need to help you find ways to attack this material better so that you can comprehend it and understand it. All right, so it's okay to not really know what's going on right now, but after 3 or 4 days of studying this material, and that's what you need to do. You can't say we're done with class and I'm going to ignore this for the next 5 days, because then it just gets worse. But if you use the next 5 days to really work over this material, make sense of this material, if you're confused by it, email me, come to my office hours, go back and look at the lecture again. Again, you don't have to watch all 4.5 hours of the lecture. You can just fast forward to the part where I'm talking about the cardiac conduction system, if that's the part that's confusing for you. Or if you that wonky slide with all of the action potential for the conductive cell didn't make sense, read it in the textbook, look at the interactive physiology, listen to the lecture again, and use those things to synthesize this information you've been presented with and make sense with it.
Professor, I have a tip.
Yes.
So this is what I did all last semester, taking your class over the summer. So I would rewatch your lectures, but I would change the speed to maybe 1.5, 1.75, and I would get all the same information in like a fraction of the time. And that seemed to help me a lot.
So are you telling me you want me to speak faster?
No. I'm just saying I already absorbed the information once.
I see. Okay, so it allowed you to go through it a second time. Yeah, absolutely. Or do it the other way, right? You can go bit by bit. You know, take a whole day to go through it, 30 minutes at a time or something like that. So yeah, work at the pace that works for you. Absolutely. And you were talking about preparing ahead of time. Well, I have a not so secret for you. All my previous lectures are on YouTube as well. If you really want to see what the third heart lecture is like, it's probably there from last, well, not probably, it is there from last spring. Now, obviously, I'm not just playing it for you again. I tend to, like I said, tinker and change things, but the meat and potatoes of it are definitely the same. So that can be something that you can do to help you to prepare as well. That's a lot of me, though. That's exhausting, but I don't even like that much of me. But if it helps
Hey,
you to
compared to the summer, having you 4 days a week, this is a cake.
Well, and so that's what I was saying, that you and Jacob the same way, right? Again, if you think about the summer, Monday, 4.5 hours, Tuesday, 4.5 hours, then you only have one day off. And then Thursday and Friday, and then only 2 days off. So absolutely, the advantage of a normal summer is you have much more time to process and make sense of the information. But the key is to make sure you do it, right? The other problem, the nice thing, you feel the pressure of the immediacy of summer school. 5 days seems like an eternity. And so there is a tendency to just, I can take these first 3 days off and just spend Sunday studying it or something like that. So don't do that as well. You can type my name into YouTube, but there's a link on our main page. If you go to our,
Be careful how you search it. Definitely be careful how you search it.
That is probably true as well. But if you go to our modules, in our modules, under the study tools, right? Class YouTube for lectures. Just click that link and that takes you right there. So you don't even have to do a search. So no scary searches. You can just click that link and that'll take you there from the modules. All right. Excellent. Great questions. Any others?
So Dr. Slutsky, this might be a better question for next week's lecture. Just curious, want to know about heart attacks and how narcotics and stimulants impacts the different components and mechanisms that we've talked about today.
Okay, so let's start easy. You are right. This might make more sense next week after we talk about the ECG, but even based on what we've known so far, let's go back to the lecture real quickly. And this, as we know, this spread of the electrical signal, this spread of the electrical signal across the atria causes the heart cells of the atria to be stimulated. When they stimulate, they produce an action potential, and they produce an action potential, they contract. And because of the flow of it, you get that uniformity to it. However, what happens is if there is a loss of blood or something like that, loss of oxygen, some other type of damage occurs inside of the cells, then what can happen is, as that electrical signal spreads, as we know, when it spreads, it doesn't immediately spread backwards because of the refractory period. However, if there's a part that is damaged, what can start to happen is that electrical signal can start to circle that damaged area. It can disrupt the signal. It can produce turbulence in that signal. And so that normal natural flow is disrupted. And if the flow is disrupted or becomes repetitive or some other type of thing like that occurs, then if you disrupt the electrical signal, you're going to disrupt the mechanical activity that is caused as an event of that. All right, so like I said, it'll be easier to see that when we talk about the ECG, but in simplest terms, it's about turbulence, right? The more rocks and logs you put in the river, the more white water you produce, the more turbulence you get, and the less effective the flow of the water. And it's the same way here. The more turbulence you have in the electrical activity, the more turbulence you're going to have in the mechanical activity, and the less efficient your pump is going to be. All right. Excellent. Any other questions? All All right, guys, like I said, we get to finish about a half an hour early, so awesome. So enjoy your weekend, study hard this weekend, and we hit everything new on Tuesday. So good luck. And like I said, if you have any questions, any problems, any issues, like I said, by all means, email me. I do have office hours tomorrow. Oh, no, I don't. Tomorrow's Friday. I can be reached by email and arrangements can be made. I'm very good at responding by emails, and I will be around this weekend, so I may be able to Zoom if necessary. All right, guys.
Oh, what about me? You said we can talk after class.
Excellent. If you still need to do the 5 for 5, you can stick around after this since we finished a little bit early, and we'll do it just right in here. I'll put anybody who's left into a weight room, and then I can meet with people one-on-one. So if you still need to do that, stick around. Otherwise, have a good weekend, and I will see you guys later.
Thank you.
Thank you, Professor.